Good morning, everyone.
Good morning.
Oh, thank you. Thank you. I appreciate that. We are having some IT issues, and there will be folks working in the background, and you may see some folks over here, so please just ignore that. We're so excited to have you all here, and I really want to thank every single one of you, whether you've traveled from near or far, for being here today with us for the methods and techniques for integrating the biological variable sex in preclinical research workshop. It's really critical that we kind of pause at the beginning of this, and I share with you where we are coming from in terms of why we chose to have this particular meeting. The name of the Office of Research on Women's Health has the research first, NIH Office of Research on Women's Health. Some of the other offices say Office of Women's Health, Office of Women's Health, whatever. The research is first in our name for a reason, because that's what we're about. That's first and foremost what we're about. And if you look at what actually the word research means, from an ophthalmologist perspective, I see all of these visual words, but it's to look again, research, to look again. And that's really what research is about, looking again, learning from what we know, learning from what we uncover, and then improving what we do based on data. So in 2014, we're at a really exciting threshold for turning another corner in research on women's health. We're nearing our 25th birthday next year, and we have indeed, I think, together as a scientific community made much progress improving women's health in a wide array of areas, but much work remains to be done. This year has been a banner year for progress. As you know, in May, Dr. Collins and I announced that NIH will be requiring applicants to report their plans to consider sex in preclinical research with animals and cells. And that's a new thing for some of us, but many in this room, that is definitely not a new thing. And many of you have done the foundational work in this area, preparing us for today. And I want to thank you for your extended effort in that area. As we work together at NIH to develop this policy and with many components, including NIH's Office of Extramural Research, one of the most important things we can do is listen and hear from our community. Since May, we have been pleased to hear from a number of researchers and scientists who are excited about what we're doing. We're here from people that said, I thought you were, why weren't you doing this all along? We've also heard from others that are challenged by the change, and we are excited to hear all perspectives and have had a request for information out on the street to facilitate that. And that deadline for that has been extended to October 24th. So please make sure that your colleagues know about that. One of the reasons for today's workshop is also for scientists and advocates to understand that we are hearing you in terms of wanting this change to be carefully implemented as a true benefit to science and not become a trivial bureaucratic box to check. I can assure you that is definitely not my priority. We're about making this better for science. We want this to be a meaningful change, and we want to implement it in a meaningful way. So today you'll hear from an outstanding group of scientists and researchers that are leading the way by routinely incorporating sex in experimental design. We can and will learn a lot from these talks, and we plan to publish these findings to more broadly disseminate this information to the scientific community. We have 4 sessions scheduled for you today. The first will address the basic concept of including male and female subjects in studies. The second will explore the impact of including or not including sex as a basic biological variable. The third will get specific practical methods to integrate sex as a variable in research plans and projects. And the fourth will walk us through how we can begin to introduce a sex matters culture across multiple disciplines. The last point is really important, that what we're doing here, everyone has a role in this change and in every area of science. This is not just a women's health issue. This is a scientific issue. This is an aspect of rigor, and this is about doing the best science possible and learning the most we can from every single experiment we do. As stewards of taxpayer money, we really can promise nothing less than that. Before we get started with the science, let me reiterate how glad I am that we're gathered here today. Sorry about that. And also I want to thank several staff who've put in countless hours executing this meeting. The entire ORWH team has done an amazing job putting together this meeting since NIH announced this new change and studying sex to strengthen science. We've been in pretty high gear, and we're a small office, which is a very big deal for us to be in such high gear for extended period of time. But in particular, I want to thank my Deputy Director, Dr. Susan Meyer, who's in the back. And one of our program staffers, Dr. Leah Miller, who's here. And of course, each and every one of you who are speakers, moderators, and for being here today. As I've mentioned, NIH leadership is fully supportive of our plans to enhance scientific rigor by considering sex as a fundamental biological variable in experimental design. And as such, as a testament to that support, I'm delighted to have with us today Dr. Larry Tabak, the NIH Principal Deputy Director, who I'll introduce in just a moment. Thanks again to all the distinguished speakers today and all of your work that you have done to get to today. We are actually coming at a really opportune time, but you have been doing this work for some time, and I want to thank you on behalf of ORWH and NIH for that work that you've done. So the purpose of this workshop is to discuss methods and techniques to learn how to integrate sex as a fundamental variable, and I like that word fundamental, and I want to keep us coming back to that along the day, and preclinical research, and through examples why that is so important to the health of all of us. I want to also just remind us in this room, as people are seeing us through videocast, to talk about the use of the words sex and gender and to use those carefully. And as each of you go, I'm sure you do this, to share with colleagues the correct usage of those terms, and sex being a biological classification and gender, of course, being related to self-representation, related to psychological traits, behaviors, and other factors. Our main deliverable at the end of today is a call to action to speakers, audience members, and those who are viewing this videocast and the scientific community. How can we continue to spread the word about the importance of studying sex to strengthen science, as well as how to do this research? Many scientists simply don't know, and we're happy to help. Throughout the day, if you have questions or comments about moving forward or about specific talks and discussion, I encourage you to write them down on an index card, which you can get from the greeting station, or to submit them electronically to ORWH Methods Workshop at sp10mail.nih.gov. I also want to encourage everyone in the audience to tweet during the day using the hashtag #SexInScience. We've had great luck communicating with various audiences, and it's really an easy way to get the word out, and it's fun. Now I have the pleasure of introducing Dr. Larry Tabak, who will talk about NIH's efforts to enhance the reproducibility of biomedical research findings and point to the fact that considering sex in experimental design is a critical piece of those efforts. Dr. Tabak is NIH's Principal Deputy Director, arguably the most difficult job at NIH. Before assuming this position, he was Director of the National Institute of Dental and Craniofacial Research for 10 years, 2000 to 2010. Dr. Tabak has provided leadership for several trans-NIH activities, including the NIH Roadmap effort to support team science, the NIH Director's Initiative to enhance peer review, and the NIH's implementation of the American Recovery and Reinvestment Act. He co-chaired working groups of the Advisory Committee of the Director of NIH on the diversity of the biomedical research workforce and information technology and informatics. As I alluded, Dr. Tabak is currently coordinating a trans-NIH effort to enhance the reproducibility of scientific findings. In short, we often wonder what he doesn't do. He's a very, very busy man, and we're delighted to have him with us here today. Before that, Dr. Tabak was the Senior Associate Dean for Research and the Professor of Dentistry, Biochemistry, and Biophysics at the School of Medicine and Dentistry at the University of Rochester in New York. Dr. Tabak's major research focuses on the structure, biosynthesis, and function of glycoproteins, and he continues to work in that area, maintaining an active research laboratory. Almost there. Dr. Tabak is an elected member of the Institute of Medicine and of the National Academies. He received his undergraduate degree from City College, his DDS from Columbia, and his PhD from the University of Buffalo. Please join me in welcoming Dr. Larry Tabak.
Good morning, everybody. So I don't even speak to my wife without PowerPoint, so we may have a big problem here. Okay, so what I'm going to do is, because I know you have a very packed schedule, fortunately I can see my slides here, and eventually you will see them when they are sent to you. But I will use the, wonderful, now you can see them. So I was going to say I would just speak, but use the slides as a guide, but we can do better than that, so that's great. So Janine, thank you so much. We have been partners in this. Very early in the process, Janine met with Dr. Collins and me, and we had, I think, a very good discussion about the gaps that currently exist. And this is a little bit like coming to a meeting where Noah is in the audience and you're going to speak about floods. You all are the experts, and so there's nothing I'm going to say that you don't already know. And so I certainly look forward to the results of your efforts today. So again, as you all appreciate, every system is tuned to get the results it gets. Unfortunately, we all fall into this default biology mode, and so in a lab that means cells derived from males, and in the clinic it's the 70-kilogram male. I only wish, but that's what we do. And somehow we amalgamate things, and but females obviously not equal to non-male, and just like minorities are not non-whites. And so in order to ensure the highest standard of rigor and reproducibility in science and science communication, we have to appreciate that the approaches and the results that are used have to be as consistent as possible and as free of biases up front. And it's only by raising the level of understanding about these issues that we can hope to achieve that. There's nothing true about the rumor that I'm going to talk about the Krebs cycle. You can all move down to the front if you like. So back in January of this year, Francis and I offered this commentary in Nature, where we sort of laid out what the NIH strategy was going to be to try and enhance reporting and rigor in scientific research. And as a result of these intense conversations with Janine, in other words, she was in our face, okay? And you can all visualize that, right? We included crucial experimental design elements that are all too frequently ignored, include blinding, randomization, replication, sample size calculation, and the effect of sex differences. And it's true. If you do surveys of the literature, frequently these key attributes are not mentioned. Now, it doesn't mean that people didn't do it, but they don't report it. And so absent reporting, you have to assume that, in fact, it was not done. And then Janine and Dr. Collins followed up with a piece in Nature, which I know has elicited enormous response from the community and some angst, and that's okay, because that's the first step, is to engage people and frame what the issues are. And then there were some blogs, and then on the director's page, Janine pointed out that ORWH and NIH as a whole is promoting a balanced approach to addressing male and female differences in both cells and animals. Now, probably each of you can tell a funny story about an investigator who comes to you and says, my cells aren't men, or my cells aren't women, they're cells, and that unfortunately underscores the work that needs to be done in this area. And many people have taken notice, both within the scientific literature as well as the lay press. My favorite is from Slate in the upper left, I guess you saw upper right. Why are all the lab rats boys? And it goes on to be even more, but it's an interesting title. Circa 2014, we're trying to reframe this discussion by articulating the problem, looking at solution, defining the action steps that we need to get there. That is requiring a deliberate approach in considering sex in cells and animals to make sure that everybody, men and women, get the full benefit of all of the medical research that's being supported. And the outcome, of course, is obvious to all of you. When researchers consider sex as a fundamental biological variable, we'll continue to deliver rigorous science that will drive medical advances. But the single most important message on this slide is that we can't do this alone as an agency, and we need to work with all stakeholders to accomplish this, which is really why you're all here. Not only to share with us the efforts of your individual work, but also to be enlisted in this stakeholder army that we need to reach out across the entire biomedical research community to let people know about this and to encourage them to move forward in the matters that's outlined. Now, there's a tremendous resource that is now available, which was put together seemingly in record time. I'm not quite sure how you guys pulled this off. So the S4 program, Studying Sex to Strengthen Science, and on this website is just a tremendous amount of valuable information. And so to anybody who is sort of interested in the first step, the primer, if you will, of how to begin to engage in this is a great place to start. And it's indeed the place that I start when I'm preparing talks of all matter, including the one I have to do next at the Center for Scientific Review's Advisory Council. Again, many of you may be aware of this and indeed engaged in this. There is a collaboration on specialized centers of research on sex differences. These are by nature interdisciplinary in their approach. And research on sex and gender factors underlying a priority of women's health issues is the focus. And of course, these are studies that bridge the divide that often exists between basic and clinical research. The investment in total has been non-trivial, and there are currently 11 funded SCORE centers, and they are listed here. And you can see that there are a number of partners throughout NIH, and that's a good thing because it ensures that the effort is distributed across the biomedical research community and is not the sole responsibility of any one organization. There's also been an effort to provide strategic administrative supplements to incentivize research on sex and gender differences. And you can see some of the many interesting topics that have been enabled by this approach. And in the bottom of the slide are listed some of the ways that this has been accomplished by adding a second group of animals of the opposite sex for comparative analysis, testing methodological issues for understanding sex differences, tests for differences in epigenetic marks, males versus females, and so forth. So, and you can review all of those because they each are enabling and help you when an investigator says, well, how am I supposed to start doing this in addition to everything else I'm doing? Here are some facile approaches that any investigator can incorporate in his or her work. If you look at the health topics, the range that have been enabled by this approach, that's displayed in this pie chart, and it's a good range of different approaches that you can see: cardiovascular and pulmonary science, substance abuse, mental health, and so forth. So it's really quite a good range. It really reflects the range of work that the NIH supports. Now, there is an online course site, and for those of you at NIH, this is not the type of course where the little bunny in the corner, yaps at you. It's an inside joke. I won't. This is a bona fide course, one that is rich in content, and for those of you who need to keep up your CME or CPE credit, you can actually get those credits through successful completion of this. Again, I'm not going to go through the details of this, but you can see these are very rich, very substantial materials. Good experimental design and reporting are what underpins rigor and reproducibility. If you can't read the cartoon caption, two white male scientists, of course, say there's a flaw in your experimental design, all the mice are Scorpios. Well, okay, it's a start, but so the obvious is stated here: the 5 requirements for good experimental design. You have to be unbiased. You have to have high precision. You need a wide range of applicability. Keep it simple, and then obviously have the ability to calculate uncertainty. And this, curiously enough, comes from something in 1958. We have an era of trainees who, if it was published prior to 2010, it doesn't matter. It's ancient literature. It's a little scary, but anyway. In terms of factorial design, we've got to get people thinking about addressing sex as a second independent variable. And this is not new. The upper left, a review article from 1992. Lower left, a review from 2003. We've known this for a long time, but yet it has not become mainstream for some reason in standard biomedical research efforts. So again, this is Experimental Design 101, something that you all know. So the basic design is displayed here. Factorial design, which gives you so much more information, is displayed here. Now, this is actually one of my favorite slide sets, and I now use it in everything that I do. So, and this is a hypothetical, of course, where you look at disease impact, and it's the tyranny of the mean, where you aggregate data, right? And so the control and the treatment arm, look sort of the same. But yet, if you stratify males and females, you can get very, very different results. And every time you show that to people, they say, that's just hypothetical mumbo jumbo. And okay, so here it is in real life. These are real data, published in 2005. The effects of the selective poly-ADP ribose polymerase inhibitor in wild-type mice of both. Well, they said genders, so I guess these were really empathetic mice and could describe things. But nevertheless, it's kind of interesting. Janine mentioned that I helped coordinate the Recovery Act for NIH. And there was always this concern because it was so visible. Everything was under a microscope. And any grant title that had sex in it was, people were thinking that we were somehow doing something that we clearly were not. And they kept telling us you have to switch sex, the word sex, use gender. Well, no, you can't, because that's not what, it really is sex. And anyway, so there it is. But nevertheless, real data shows that this can happen. And this is sort of burned in my brain now, and no doubt in yours as well. And who knows how many other examples of this. And so you say, well, this is about mice or rats, but when it comes to humans, and you think about intervention, and that's where, of course, the rubber meets the road. So lots and lots of folks are intensely interested in this, and that's a good thing. Both members of the Congress and here at NIH, where there have been a number of policies that have been elaborated, and Janine has already spoken about the RFI. And as part of the overall reproducibility effort, we convened an interesting workshop with journal editors from preclinical journals, and it was somewhat arbitrary. We invited editors of the top 35 journals that NIH-supported investigators publish in. So whoever those were, that's who we invited. And among the things that we discussed, and imagine getting 35 journal editors in a room trying to get them to come to consensus about anything. But among the parameters that we discussed was the need to report sex in the work that was being published in their journals. And indeed, they eventually adopted this as a principle. There will be joint editorials published by Science and Nature and maybe other journals as well in early November, so stay tuned and you'll hear a little bit more about this. Many efforts accounting for sex differences. One that can be discerned from this article in Nature Biotechnology as part of the so-called COMP project. This is a systematic ablation of every gene in the mouse genome. They did broad phenotypic screen of 472 so-called knockout lines, and the results are reported for male and female aggregated, male alone, female alone. And so it's a good resource for people to turn to. NIA has been doing a fair amount of work in this, and I'm just giving you some examples. Obviously, time precludes me from doing an exhaustive list, but NIA has long had this testing program, and along the way, they have come up with some interesting things to look at a series of interventions that are purported to expand the lifespan, and they are pretty rigorous about this. They use male and female genetically heterogeneous mice. They use a four-way cross design. And they always use sufficient numbers of male and female mice to test 80% chance of detecting increase or decrease in the lifespan with a threshold of 10%. And so in the box in the upper right, you can see some things work for boys and some things work for girls. Now that's a big surprise, but nevertheless, it typically goes unstudied, not unreported, unstudied. And again, you all know this, but it certainly is important as evidence as you speak to colleagues who may not be as knowledgeable about these issues as you are. And then there's the whole subject of, what's the sex of your cell. Actually, first figuring out who your cell is a good first step, because there's some very scary stuff going on. And NIGMS is leading an initiative to try and clean that up. Recent analysis of the RNA-seq database has shown that about 11% of the cell lines are contaminated with mycoplasm. So whether the cell is a boy cell or a girl cell, if you're contaminated with mycoplasm, that's going to be an important thing to understand. Nevertheless, once you get past that, knowing the origin is quite important, and it's important to periodically review this because of issues related to genomic instability. So something that started with a Y chromosome may not be the same as it keeps being passaged and then passed from lab to lab, and we all know how that happens. The reporting, of course, is crucial, and a workshop summary of the IOM laid out some of the principles of this for preclinical studies, and I won't read this to you. You can obviously do that more rapidly. And also for clinical study design. And again, this IOM report is a superb resource for those beginning to enter this field, or for those of you who are advanced practitioners but need some ammunition to convince some of your more intransient colleagues. And then clinical study reporting is located here. So, again, if you consider this paper from BMJ back in 2002, does animal experimentation inform healthcare? Well, it can, but if and only if it's done in the most rigorous, transparent way possible. And I think it would behoove us all to keep this in mind as we try and cross that extraordinary divide from the animal, the so-called preclinical model to that first-in-human trial. There are all sorts of implications, but unless you overcome the issues of bias, sample size that may be insufficient, we don't advance things. A number of options have been considered to ensure more rigor in this particular space, drawing on what we do in human clinical trials. The notion that you would do a pre-registration of the trial with endpoints clearly articulated, instead of what, and not that anybody in this room has ever done this, but instead of what occasionally you see somebody running around from statistician to statistician, make my results significant. Have any of you ever seen? I see a few smiles, so I guess one or two of you have observed this, something, of course, to keep in mind. I think the most important thing of all, and it's something that Janine obviously has already highlighted, but I'll reemphasize. As a starting point, we need to speak about these issues. This is not NIH, saying you will now do the following. This is about NIH using the power of convening experts to hear as wide a range of viewpoints as possible, to begin to develop rational and scientifically justifiable approaches to enhance what we're trying to do. And there are two, I think, very excellent pieces that have appeared recently, which articulate many of sort of the initial points that no doubt form a framework for this. And I think workshops like this one will extend the good work that has already preceded, as summarized in these two reviews. So with that, I will thank you for your attention and turn the podium back to Janine, for what I think will be a great workshop. So thank you all very much.
Thank you again, Larry. I really appreciate you being here. I think I can speak beyond the walls of the Office of Research on Women's Health to say how much we appreciate the leadership and support that Dr. Tabak has provided, along with Dr. Collins, in trying to push forward the development of policy to considering sex in preclinical research. Now I'm happy to introduce our first keynote speaker, Dr. Larry Cahill. Among this group, he hardly needs an introduction. and as he's been a champion of the need to study male and female biology for many years. Dr. Cahill is a professor of neurobiology and behavior at the University of California at Irvine. He is a neuroscientist focused primarily on memory. Dr. Cahill first became interested in brain and memory as an undergraduate at Northwestern University after working for 2 years at Searle Drug Company in Illinois on memory-enhancing drugs. He earned a PhD in neuroscience from the University of California, Irvine in 1990, then conducted postdoctoral research in Germany for several years. Dr. Cahill came back to UC Irvine to extend his research to studies of human subjects, which in turn led to his discoveries about sex influences on emotional memory, and to his current general interest in the profoundly important issues of sex influences on brain function. His research suggests a central role for the amygdala in human memory formation under conditions of emotional arousal. Along with being a long-standing leader in the area of brain and memory, more recently, he has become an influential leader on the topic of sex influences in the brain. Dr. Cahill is an internationally regarded dynamic speaker whose work has been highlighted extensively in the press, including the New York Times, the London Times, PBS, and this year on 60 Minutes. Dr. Cahill's work is strengthening the notion that sex and the cerebral hemispheres are twin interacting influences on brain mechanisms of emotion and memory. His talk today is Sex Influences on Body and Brain, an idea whose time has come. Please join me in welcoming Dr. Cahill.
All right, thank you very much. We're running a little late, and I'm going to do my best to stop at 9:15 because we have a packed schedule and a lot of great people with a lot of things to say. First thing I want to say to Janine is thank you very much for inviting me to be here. I am thrilled that this meeting is happening, thrilled that I was asked to speak, and thrilled that I feel relatively cognizant at 5:30 in the morning California time. I thought I'd start with an anecdote. About 15 years ago, when my work on emotion and memory first drew me into this topic, I attended a seminar out at UC Irvine. A woman was talking about her work on stress and the hippocampus, stressing rodents and seeing what it does to the hippocampus on a cellular molecular level. And this work, she said, was going to have a lot of impact on understanding two disorders in particular, clinical depression and post-traumatic stress disorder. Having just gotten into this issue, I raised my hand and I said, "Are you studying male or female rats?" And her answer was, "Male rats. I don't go near female rats." And when I pushed her on this, she said, well, the basic answer was that the nice story she was developing in the male rats wasn't really holding up in the female rats. And so I realized at that moment that she was operating under the conception that a lot of people were operating under, that even though her results, by her own admission, were not generalizing from the male animal to the female animal, she was freely generalizing from the male rat to human conditions, and in fact to human conditions that mostly affect women. Shortly thereafter, I was at another talk at Irvine, and I was talking to a young graduate student whose lab was studying both male and female rats because they had them in the vivarium, not because they cared. And he told me that it was an auditory physiology lab, and they were finding what they had been expecting to find in the male rats. But he told me that the things were not working out in the female rats. It just wasn't working out. I said, "That's really interesting. What are you doing about that?" He said, "Well, now we just kill the females at birth," which struck me as an interesting way to deal with the sex difference issue. In fairness to them, he was basically saying, we don't know what to do with this, and we can't afford the vivarium costs, but these examples illustrate the kind of issues we've been dealing with sex differences for some time. This year, as Janine mentioned, with this wonderful article by Janine and Dr. Collins, we've seen some terrific changes. So my goal is to just capture some of the importance of this issue and why this is not a bad thing. This is a wonderful thing that's happening to science now. By way of a little background on me, I have studied for about 35 years the neurobiology of emotional memory. Framework shown for you here. The basic idea is that stress hormones activated during and after emotional events feed back to this amygdala, and through its immense connections with the rest of the brain, the amygdala tells other parts of the brain, crank up the memory, that was better. Well, about 20 years ago, after using exclusively male animals to study this, I shifted to studying the human animal. And when you do that, you naturally start to study this thing called the female. And in doing that, I stumbled upon something that was really a career changer for me, a sex by hemisphere difference in the relationship between the amygdala and emotional memory. And I probably won't have time to go into any of my own work on emotion and memory, but it was a game changer. And what it did was it drew me into the general issue of sex differences in the brain, and I basically started to educate myself and realized that I was operating under a bunch of false assumptions. So what I'm going to try and do in my time that I have here is to capture, again, why this issue, I think, is so profoundly important, why it's so wonderful that these changes are taking place now, and I'm going to do that by addressing 5 questions. First, although a lot of people in this room may not need convincing, I'd like to address the question, is the degree of sex bias in animal and cell research really all that bad? Is it really that male-dominated in general? And my answer is going to be yes. Then I think it's important to spend some time thinking about how did we get this way? How did the status quo evolve? And what are the sources of resistance that have been preventing this issue from moving center stage where it belongs? So I'm going to talk a bit about that, and I'm going to suggest there's several key reasons. Having done that, I want to briefly raise the issue of, so where are all these sex differences? And the answer is pretty much everywhere, and I'll focus on my area of brain science to give you some examples of just how ubiquitous, and really, I really mean ubiquitous, sex influences in the brain are. Does all this really matter for the clinic? At this point, it probably will be self-evident for people that the answer is yes. And what should we do about it is the last question. I won't spend too much time on that, as that's really the point of this exercise. How do we go forth rationally on this topic? So first, is the degree of sex bias in animal and cell research really all that bad? Couple things to say. First, a wonderful paper by Irv Zucker at Berkeley several years ago covered the literature, 8 or 9 different fields very well. And adding it all up, there was a ratio of about 5.5 to 1 male to female animals, far worse in some domains, reversed in some other domains where they primarily use females. But overall, a very clear documentation of the bias towards the male. More recently, Melina Kibbe and her colleagues at Northwestern, I think this was just a couple months ago, they did a similar thing in their domain of surgical research. They took a look at the top 5 surgery journals and summarized everything they could find for 2 years, all the animal and cell research, and asked the question, who's studying males and who's studying females? And the answer is almost everybody's studying males. Within cell research, mostly they found that people don't even report the sex of the cells, and it's because it's really not on people's radar screens yet that this should matter. But when they do report the sex of the cells, there's a tremendous bias towards the male. And naturally, if you just count up how many males and females are studied, there's an extreme bias. And I think most intriguing and kind of disturbing, really the reason we're here, is even for female prevalent diseases, of those reports that stated sex, only 12% studied female animals. And, my view is that even if that was a justifiable approach some years ago, it is no longer. The last example of sex bias in the numbers of animals comes from my home turf, learning and memory, where we may be the worst offenders, as evidenced by this graph from Mohammed Milad and Kelmer Lebron Milad from Harvard a couple years ago, and the data pretty much speak for themselves. I never saw a female animal in all my years of studying brain and memory. So is the degree of sex bias in animal cell research, is there really an issue here, in case anyone who's here in the room or watching has any doubts? The answer is yes, there's an extraordinary bias. So how did we get this way, and what has been the sources of resistance to studying both sexes, studying sex influences? And I want to hit on just what I think are some of the biggies. The first is this idea that females are just too variable and complex, and one has to deal with the menstrual cycle, the estrous cycle. It's kind of an ironic thing to say because we're supposed to be helping females in the real world, and last time I checked, they had menstrual cycles in the real world. But it's an idea that has been out there, and is it true, for example, when it comes to animals, that the females are more variable because of the estrous cycle? Again, back to Irv Zucker out at Berkeley, a wonderful study that came out this year, where he said, let's look at the variance in male and female rodent studies. I forget if it was rats or mice. Let's just look at the variance. in males and in studies involving just females who are cycling, female rats. And the purple area on the bottom there shows the degree of where there was overlap between the variants in the males and the females. So in this one powerful paper, Irv and his colleagues have blown out of the water the idea that if you have to study the female rat, you have to deal with estrous cycle effects. This is not to say there are not estrous cycle effects, but as a first pass, it's not a reason to not study females. But here, I think, is this next source of resistance might be the most important, at least in my view, from hearing everything for the last 15 years. And it gets back to this issue that Janine mentioned, and I think also Dr. Tabak, on fundamental. I was having a discussion 3-4 years ago with a colleague at Irvine, an fMRI guy, about sex differences, and he said, it's not that I'm not interested in sex differences, it's just that I want to understand the fundamental mechanisms first. And then he kind of caught himself because he just basically said my work is not fundamental. But I'm glad he said it because it was a light bulb moment for me where I realized that's the problem. He views sex differences as not fundamental, something to be gotten to later, after we figure out the fundamental mechanisms. And I happen to teach a history of neuroscience course at Irvine. I think it's the only history of neuroscience course in the United States of America. And I started to think about this from a historical perspective. Where does this come from? And I started to realize that it's this assumption, which is starting to crumble today, in our time, that the fundamental is the shared. That the women in this room and who are watching this, what they share with the males is what's fundamental. And what conversely they don't share is not fundamental. And we'll figure it out later. And where does this come from? And I started to realize it's got about a 500-year tradition of us assuming the fundamental is the shared. Go back to Western science finding number 1 about how our bodies work. The thing we figured out that we still believe today, the idea that the blood goes in a circle. Nobody at the time said maybe it's not true in women. When Galvani and Volta and others argued over a 200-year period about whether we are electrical as opposed to having spirits flowing up and down our nerves. And they finally decided, by gosh, we're electrical. Nobody said, we're not sure it's true in women. When Cajal told us that the brain itself, cell theory applies to the brain. Weird cells, but cells nonetheless. No fundamental discovery. Nobody said, well, we better check it in females. When Watson and Crick gave us the double helix, nobody said, I wonder if it's a triple helix in women. So I'm being partially facetious, but I'm making the point that we have a deeply ingrained 500-year tradition of believing that what's fundamental is what we all have. That's why it's fundamental. But what's happening, of course, is we're starting to realize, especially in the last 20 years, that males and females are fundamentally similar and fundamentally different. And it's the fundamentally different part that needs to be studied. Another source of resistance, and I apologize if I fly real fast, but I want to make sure I end on time. Sex is just another, this one I've heard quite a bit in the last several months. Well, sex is just another one of many variables that can influence our outcomes. So why do we care about sex so much? And my answer to this is, well, of course, sex is one of many variables that can influence our outcomes. We all know that. But it's hardly just another variable, because there can be no other variable that influences a greater percentage of the population. There can be variables that influence less. There can be no other variable that is more evolutionarily fundamental. There can be ones that are less. And there is no other variable for which we already have such an abundance of evidence indicating its importance. And yet, the degree to which it's studied remains so relatively low. But to those who insist that sex is just another variable, I would like to point out that I don't think we treat it like just another variable. And to make that point, I'll show you these numbers I put together from various organizational structures in the NIH that I could find that were devoted to well-known individual difference variables. The variable on the top is NICHD, the variable of being a child or developing. This is the budget from last year. We clearly think this individual variable is important to the tune of about $1.2 billion a year. There's the variable of getting older, aging. We think that's important to the tune of about over $1 billion a year. There's a variable of being in a minority race. I think Dr. Tabak mentioned this. It's a relatively young institute, still scratching and clawing for its piece of the pie, $270 or $80 million a year. And there's the Office of Research of Women's Health. So my statement to those who insist that this is just another variable is, so let's treat it like just another variable and start spending, call it another $1 billion or so a year on it. But clearly we're not treating it like it's just another individual difference variable. And again, I don't think it is, and I'm neither stating nor implying that I think there should be a National Institute of Women's Health. I think that would be a disaster because this issue is far too big, too important to be relegated to a single institute. Another chief source of resistance, this will be the last one I mentioned. We heard of Dr. Tabak mention some sort of politically correct statements on doing this. The truth is that political correctness has worked powerfully against studying sex differences since at least the 70s. There are still people who refer to me as a neurosexist because I'm actually interested in what Mother Nature thinks about the differences between males and females. You may not have heard of this author, but you've heard of Gloria Steinem, who was quoted as saying, it's really a remnant of anti-American crazy thinking to do this kind of research. So the idea that this is being driven, what we're experiencing now is being driven by political correctness is, I think if you think that, you haven't been paying attention. Because in fact, it has been political correctness, especially for research from here up in the brain, has been a powerful force against studying sex differences. And personally, I've come to believe that this is anchored in a false assumption. An assumption that if two groups are equal, then the two groups are the same. And conversely, if a scientist shows that males and females are not the same in some respect, they are showing that they are not equal. And that's false. It's just false. Because the truth is, equal does not mean the same. And if you're interested in this, I published an article you can get online at the Dana Foundation addressing this. But the fact is that if two groups really are different on average, but they're being treated the same, then they're not being treated equally. And that's exactly the case in research in medicine today, where women are not being treated equally with men because, by and large, they're being treated the same. So these are some of the sources of resistance that I think has kept this issue stuck in a corner. I believe in pulling these things out, hitting them head on, and then asking people, do you share this view? If you do, defend it or abandon it. And that's why I like to address these sources of resistance. So, having done that, where are all these sex influences that everyone's talking about? And the answer is pretty much everywhere. I'm going to focus again on neuroscience because that's what I do, and give you some examples of how ubiquitous sex influences are. And in order to explain it to you, I need to first explain how we used to think, or at least I used to think about sex differences in the brain, being trained in the 70s and 80s. This is what sex differences in the brain meant to me. Well-known article by a founder of the field of behavioral endocrinology, Seymour Levine, article from 1966 entitled Sex Differences in the Brain. And the whole thing boils down to one structure, the hypothalamus. And for me and for many neuroscientists who didn't study this, sex differences in the brain basically meant the hypothalamus, sex behavior, sex hormones, stop. Wasn't the hippocampus, wasn't the amygdala, wasn't the caudate, wasn't the neocortex, wasn't anything that we needed to concern ourselves with. The reality, and it's really exploded in the last 15 or 20 years, is that sex differences are everywhere, or sex influences, I like to say, probably a more accurate term, sex influences. From the intact functioning human to neurochemistry of the human to primate behavior to knockout mice. The knockout mice people are interesting because they don't just order male animals like the rest of us used to. They make mice, and they make male and female mice, and the mice are too expensive to throw half of them away. So they're discovering sex differences all the time, and sometimes they publish the one that makes sense and put the other one in a file drawer. But they're all aware of this, so it's really a driving force. The hippocampus, as I'll show you in a minute, an example is sex difference, and there's even sex differences in neurons in petri dishes. A couple examples to illustrate the power of this issue. This from a colleague of mine out at Irvine, Frances Leslie. She's interested in what happens when mothers smoke. So she takes female pregnant rats, she exposes them to nicotine. Then when the pups turn into rat teenagers, she takes a look at their brains for the expression of genes. In this case, you're looking at about 15 genes related to myelin, or the white matter. And the blue bars show the animals that were exposed to nicotine in utero. White bars were not exposed to nicotine in utero. So what's her conclusion? Her conclusion is, wow, exposure to nicotine in utero dramatically and consistently elevates the expression of genes related to myelin production. What does that mean? That's the next question. But it's a pretty dramatic effect, isn't it? And that would be Frances's conclusion until she studied the females. And she found this. Now, Frances was wise enough to separate the animals, to not say sex doesn't matter, so just pool them. Had she done that, what would have happened? She would have had a whole bunch of nothing, a bunch of negative effects. She would have been wrong twice about the effects of nicotine on the developing brain. Now, importantly, this is a really important point. Frances doesn't know why this happens. I don't know why this happens. You don't know why this happens. Does that mean we get to dismiss it? Because I hear that one a lot. Well, we don't know what this means. So the truth is it's just the opposite, right? We don't know what it means, therefore we need to study it. Right? And Frances told me she got a lot of grief from reviewers and editors in publishing this because, we don't know what it means. Blah, blah, blah. It's so silly. We don't know what it means, therefore wake up and start studying it, is my view. Another example of sex differences that are striking, Louise McCullough, who was mentioned earlier, the current president of the OSSD, has been amongst the leaders demonstrating that neural mechanisms of apoptosis, programmed cell death, are both similar and different in males and females. Being dependent on the enzyme caspase in one sex, the females, and being independent of it in males. This is striking. You start to think about how you want to treat and prevent cell death. Don't you necessarily have to think about this key enzyme and its different roles in males and females? Catherine Woolley from Northwestern, to take us to the hippocampus, has some new data that she allowed me to share here. Differences in essentially the effects of an endocannabinoid, endogenous marijuana, in the hippocampus. She has found, basically, she studies the synaptic transmission in the hippocampus. And in fact, in this example, it has about GABA, or inhibitory transmission. And she studied the effects of something called a Fa inhibitor, which blocks the breakdown of the endocannabinoid. And essentially, she found that this Fa inhibitor was doing nothing to synaptic transmission, GABAergic transmission in the males. But it was profoundly reducing the effect in females. And the way she found this was she started out in the females looking for an effect that wasn't supposed to be there. This drug was not supposed to be doing this because the literature said it doesn't do this, said it very clearly. The literature was all males. So Catherine, in this now new data, has found that there's this striking sex difference. This isn't a little effect. So here we are, folks, in the hippocampal synapse, the holy center of the learning and memory field. And here's one example of an absolutely striking sex difference. And should we ignore it because we don't know what it means? Of course not. It's incumbent on us to pursue it because we don't know what it means. Is this true in humans? Absolutely. A couple of favorite examples. The gene that more than any is associated with risk of getting Alzheimer's disease. Every Alzheimer's disease researcher knows it, ApoE4. Well, it turns out it's not really true in males. A fellow named Michael Grecius and his colleagues at Northwestern have been picking up what was a 20-year-old and forgotten story, which holds that basically it's really bad to have this form of this allele if you're a woman. It really doesn't matter in men so much. Michael says that he's not even sure he would tell a man who has this form of this allele to even worry about Alzheimer's disease. And think about this. This is the gene that more than any other is associated with Alzheimer's disease, but it's probably not true in men. Yet the Alzheimer's field continues to, by and large, ignore this. There are sex differences in the white matter connectivity of the brain, which are striking. A paper from last year, a huge, impressive paper from Penn, Reuben Gur and his colleagues found just very intriguing differences in the degree, in the pattern of connectivity, with the male brain being more modular in its connectivity and the female brain connecting more across regions, especially across the hemispheres. And this fits with a well-established literature, goes back to the 70s, that holds that by and large, as a general statement, the female brain tends to be more bilaterally organized and the male brain more asymmetrically organized, as shown in this example here of people responding to sounds. With an MEG study, you can see a much more bilaterally focused response in the female brain and a more unilaterally focused response in the male brain. So there are sex differences all over the place in the human as well. I think in the interest of time, I'm going to skip talking about my work. Is that allowed for a scientist? Suffice to say that my studies of emotion and memory and this amygdala effect drew me into it. And really, it's been very refreshing. We found sex differences in the amygdala at rest. We found sex differences in the impairing effect of beta blockers on memory, affecting gist versus detail in men and women. We found enhanced memory for details in women who are at the mid-luteal high hormone phase of their cycle. And we've even found that hormonal contraception alters the retention of gist and detail in women compared to in women on the pill versus naturally cycling women. All of this, it's like there's been a flurry of exciting findings in the last 15 years since we dropped the implicit assumption that sex doesn't matter and adopted the assumption that it can and probably does. We've even found recently that what's called the global advantage, the ability to see the top left corner there, the ability to see the S over the F, fluctuates in women across the menstrual cycle in a way that's consistent with our gist detail findings that I just flew over. So this is really intriguing stuff, and that's all I'm going to say about it. I do want to say, before I get to my next topic, that I only started to read Darwin a couple years ago, and it was an effort to understand why are all these sex differences popping up everywhere. And I realized in reading the man, who's just brilliant, that he did not believe that evolution was driven by natural selection. And most people go, huh? And it's absolutely clear that Darwin felt that evolution by natural selection alone would fail. What Darwin said was that evolution was driven by two independent forces, natural selection and sexual selection, the first acting on the basis of whether or not an animal lives, the second acting on the basis of whether or not an animal makes a baby. And Darwin knew that he needed both for his theory to survive because he knew that there was too much that his theory of evolution by natural selection could not explain, most famously, this guy. He knew that, according to natural selection, this guy shouldn't exist. His grandson said that whenever he saw one of these in an English garden, it made him nauseous. So why does he exist? Think about it. This is a great way to get selected out, isn't it? So why does he exist? According to Charles Darwin, it's because she likes it. And I like this picture because she doesn't look like she likes it all that much. But that was Darwin's view, and we're having fun with it, but he got a lot of grief for this concept. And in the introduction to his second edition of his second book, Descent of Man and Selection in Relation to Sex, he said, "My conviction in the power of sexual selection remains unshaken." Why is this important? Because when you start to conceive of evolution as a complex mix of natural and sexual selection forces, like Darwin did, sexual selection is a force that by definition often acts male against male, female against female. It is therefore a force that must produce sex differences of all sizes in brain and body, as Darwin discussed in great detail. And so if you start to think of evolution as Darwin thought of evolution, you start to see that sex differences are not some weird conundrum. They're what we should have been looking for all along. I also want to make sure before my talk is over that I emphasize to those in the room and those listening that while I emphasize the biological, I get that there is the cultural and that we must understand the cultural and the influence of culture. As one example, here's a wonderful study from a colleague, Marina Pavlova at the University of Tübingen. She had men and women do a simple task. Organize a bunch of pictures into a story. It's called the event arrangement task. If you look on the left there, I don't have a pointer, but the bars on the left, the green triangle and the red circle, that's men and women just doing the task. And you can see they do it equally well. One bar over is what happens if you tell people prior to their doing the task, if you say, you know, women are usually better at this. Women's performance goes up a little bit, men not. Next bar over, info 2, is what happens if you tell both the men and the women prior to doing the task that, you know, men are better at this. The men's performance goes up a bit, the women's tanks. And then last, if you say men are usually worse, the men's tanks. So what is this saying? Is this a biological thing? It seems not to be very biological, doesn't it? So I get that there are biological influences that are interacting with cultural influences, and I'm getting my 5-minute warning. So does all this really matter for the clinic? The answer is absolutely. I'll give a couple examples, again, from my domain of some papers that got a lot of press. Here's one: post-traumatic stress disorder is associated with PACAP and the PAC1 receptor, a pituitide receptor. A lot of attention, written up in the New York Times. paper that said, wow, here we have a possible new way to treat post-traumatic stress disorder. But the truth is that it simply wasn't true in men. So all the talk was about how great this is, but it's not for men because it simply wasn't true. It was only true in women. In fairness to the authors, they put that in the abstract. I'm going to suggest it should be in the title, but they put it in the abstract, which is fine, but still it gets blown away as people discuss the implications of it. Another example, more recent, epigenetic modification of the glucocorticoid receptor gene is linked to traumatic memory and post-traumatic stress disorder in genocide survivors. This is a very good group of investigators who took a look at Rwanda genocide investigators and found that the epigenetic modification of this glucocorticoid gene changed their risk. The only problem is it wasn't true in women. It was just true in men. So same thing, reverse situation, but the same thing. If you forget this, you're in the way to making a large mistake. And yes, they mentioned it in the abstract, but again, it gets lost in all the discussion in the press. I want to briefly mention something that I'm working on that illustrates that this is also not just a women's health issue. There was a class of drugs called Lazaroids. You haven't heard of them because the FDA didn't approve them. The Upjohn Company spent millions and millions of dollars developing these drugs called Lazaroids, literally after the biblical figure Lazarus. So powerful were they in preventing brain damage after stroke. Why isn't anybody taking Lazaroids today? Because they weren't approved. Why weren't they approved? Because it only worked in men. And the FDA didn't buy the argument of approving it for men and women in the hopes of helping men and hopefully helping some women. No one thought 20 years ago to improve it for men. So think of how many lives have been lost and people have been damaged because the idea was that something's wrong because this drug is not working in women and it should be. Think of how many lives have been lost because of a false assumption. So does this matter hugely for the clinic? You're darn right it does. Is it just a women's health issue? Of course it's not. It's much bigger than that. What has this resulted in terms of the FDA giving sex-specific instructions? Not much. To my knowledge, there is at this point one drug of all the drugs out there that the FDA has not suggested. They're very good at suggesting things and guidelines, but there's one drug where they have mandated that the makers give different doses for men and women. One, and it's Ambien. And it's about time for me to wrap up. Yay, I got 2 minutes. What should we do about this? Well, that's what this conference is about. And again, I'm thrilled that this is existing. I want to make 2 suggestions. The first is nothing's going to change until the next generation changes its thinking. And there I want to highlight the work of people like Virginia Miller and Marjorie Jenkins at the Laura Bush Institute for Women's Health in Texas. These people have been amongst the leaders in creating medical school curricula. I encourage you to go check out this website that takes this and makes it very practical for students trying to learn things. This is wonderful and often unheralded work, but this is really what's going to change things. When the next generation thinks, well, of course I need to consider whether it's a man or a woman. I would be a fool not to. And that's going to start with work like this. Lastly, I want to make one suggestion, my suggestion for how to go forward, and it's in something I've been advertising, something I've been suggesting for a couple of years. I call it the Clarity About Sex rule. As many people know, you go to the literature, you often can't even find if it's a male or a female in the paper, or if it is, or they didn't do the analysis. And I've been saying for a couple of years, including to the editor of the New England Journal of Medicine a couple of years ago, who didn't like the idea, that in all papers that involve only males, the last 2 words of the title should be in males, except for things like prostate cancer. And all things that involve females, the last 2 words of the title should be in females, except for things that involve ovaries and whatnot. I see no reason not to do this. Costs not a penny. Will go a long way to clarifying the literature, which is a wonderful step right there. Sometimes you can't even find out. And to raise consciousness of this issue's importance. Well, I made exactly this suggestion a couple of years ago. I was here at a meeting at the NIH. I made the suggestion to a fellow who was one of the institute heads, one of the 27 institute heads. I forget his name. We were having a nice chat about how important this sex difference issue was, and he was saying, yes, it's very important, we need to work on it. And I said, well, would you be willing to adopt this rule for your institute? It won't cost a penny. And I remember his answer, this is a quote, he said, well, I wouldn't go that far. And, my attitude is it's Time for him and all of us to go that far and further. The evidence makes abundantly clear now. The last 20 years has been remarkable. No matter what assumptions we operated on since Thomas Willis and the circulation of the blood, the evidence has made abundantly clear that the answer to the question of are males and females same or different is yes. That they are same in many respects, different in many other respects. The different part remains today, this minute, massively understudied. And it is to the detriment of the health, not just of women, although arguably primarily of women, but of, as the Lazaroids example shows, to the detriment of both men and women. It's time for it to go. So I want to close then with a quote from a fellow whose intellect I think is far greater than mine, the wonderful novelist, writer, and poet Victor Hugo, who said, there is one thing stronger than all the armies in the world, and that is an idea whose time has come. Sex Matters is an idea whose time has come. I want to say before this is over, thank you to Janine, the Office of Research in Women's Health, Susan for putting this together, and to Janine and Dr. Collins for your game-changing article in May, and thank you again to all of you for listening.
Thank you, Dr. Cahill. We'd like to entertain a couple questions.
I'll entertain the questions. I won't necessarily answer them. Yes?
That was a fantastic talk. Bravo. I wonder if... Could you hear bravo?
I always hear bravo.
I wonder if you would be amenable to a corollary to your sex rules. Is that what it was called?
Clarity about sex rules.
Clarity about sex rules. And that is that you asked that the NIH and all their press releases indicates the sex of the animals. I've always remarked that the press releases have similarly had a lack of description, and even since the landmark paper was published, the press releases from the National Institutes of Health still lacks that fundamental variable, which again would cost nothing. It would be a great corollary.
I'm in complete agreement. I think we should start with every grant coming into every institute of the National Institute of Health starting tomorrow. Can that be done by tomorrow? Needs to say this. And why not? It doesn't cost a penny. I said this idea to the editor of the New England Journal of Medicine a couple years ago, and he did not like it. But I think if the NIH leads, they're all eventually going to follow. And I'm wonderful to see that this is now happening. No more question entertaining. Thank you.
At this time, we invite our moderator and speakers for session 1 to come up. Our speakers can sit here in the front row. Please note that each presentation for the speakers will be approximately 15 minutes, and we have designated 5 minutes at the end for questions. We ask that you hold your questions to the end of the speaker's presentation. That's what I was going to say.
Good. Are you going to put them up for slide? Are you going to put them up and think about the speaker should I? Okay, great.
Good morning and welcome to session 1. It's my privilege to be invited to moderate this session because, at least in the National Cancer Institute, we are very concerned that the research that we do from very basic science all the way through the clinic involves both sexes and genders, and that this work be done rigorously. In this session, we'll explore the concepts of how important it is for research progress that we include both male and female subjects, whether they're animals or their cells, in preclinical experiments. Some of the ways that there are to perform effective studies that have both sexes, what methods there are to account for variability, and ways to incorporate sex as a variable in experimental design. through 3 presentations from 3 very highly qualified individuals. This session will portray the rationale for including both sexes in preclinical research, present how incorporating both sexes enriches the knowledge base from preclinical research, describe various experimental and statistical considerations for research designs that include both sexes, and provide several effective examples of how such robust experimental designs can improve our understanding of sex to human biology and its representation in animal models. My own interests in this area are in the context of cancer models. Having managed for the last 15 years a research consortium designed to generate state-of-the-science cancer models in mice, and to learn how to use them effectively and reliably for cancer research. Recently, the NCI phased out this consortium not because it wasn't highly successful, but because we needed to move on to ensure that reliable use of cancer models was incorporated across the spectrum of science in the oncology community. So now we are starting an open oncology models forum to engage the many interests that the oncology community has in animal models. We're enabling a collaboratory concept to flourish. A collaboratory is transdisciplinary research performed without the confines of brick-and-mortar institutions or even the borders of nations. To draw together those who generate and study animal models, those who use them for preclinical and translational research, and those who are actually trying to decide whether animal model use is appropriate for the kinds of questions that they have. For this Oncology Models Forum, the hardware, middleware, and software platform we're employing enables collaborative work worldwide so that everyone can, in a single private space, share data, analytical tools, images, and other large files in real time without actually downloading them to their behind the firewall systems at their institutions. In cyberspace, the oncology community will be able, from their iPads or even their iPhones, tackle together the challenges of ensuring that when animal models are used for various aspects of cancer research, that the results will be thoroughly described and documented so that others can reproduce these studies, and the documentation that will be housed on this platform will lead to oncology community-based best practices and standards. As I said at the beginning, incorporating sex as a variable in preclinical and translational animal research is a prominent need in cancer research, and so we support this effort at the NIH level to encourage more robust experimentation. So without further ado, we'd like to start off this particular session with a presentation from Jill Becker, who's from the University of Michigan. Go Wolverines. It's entitled Sex Differences in Preclinical Research.
Well, thank you, Cheryl, and thanks to Janine for putting this session together. Oh, good, a pointer. And I wasn't really sure what to talk about because I've been thinking about sex differences for so long. It sometimes doesn't. It seems obvious to me. But I was so delighted when the this commentary came out in Nature that it really made me start thinking about how would I think about sex differences if I wasn't already doing this? And what is it that sex differences really can contribute to help us begin to understand fundamental differences and effects in biology? So I know that one of the reasons that we've been supported for looking at sex differences now is because of the concern about variability in data that Dr. Tabak talked about. And so I started thinking about, okay, how can we think about variability in data? So we know that, I can figure out how this works. Well, variability can come from experimental issues that are caused by the experimenter or not being very careful in how our data are collected, as well as things like testing conditions and housing conditions. But those are all things we can control. The things that we're interested in are actually what causes certain individuals to be different. And sex differences and individual differences go hand in hand in thinking about how we can better understand the biology. So the problem is what we mean by sex differences isn't always inherently obvious to everybody. I know I've heard Dr. Cahill talk previously, and he would say that when he said he'd seen sex differences, people would say, yes, but there's more overlap than, between males and females than there is in the difference between them. And he would always say, yes, but if there is a difference, then a statistically significant difference, that means it's biologically important. But there are a lot of people who think because there's so much overlap that the sex differences aren't really important. And other people who simply want to think that sex differences are bimodal. The females are like this and males are like that. And we know that neither of those are always exactly the same. So what do we mean by sex differences? So I think we need to think about sex differences as not being just one thing. And that's because we have qualitative sex differences in some things, quantitative differences, differences in the incidence of things between males and females, as well as mechanisms and combinations of these. And I'm going to give examples of each of these to illustrate this point. So for qualitative sex differences, in this case, this is something that we probably wouldn't study both males and females in. Okay, so in sexual behavior, males mount and females exhibit lordosis. So if you're studying lordosis behavior in the female, you don't need to do males. And if you're studying mounting, similarly, you don't need to study females. There's huddling behavior in female rats is also something that primarily females exhibit, although it is possible to get males to show, male rats to show huddling behavior if they're exposed to pups for a considerable period of time. So if it is a qualitative sex difference, one doesn't necessarily need to include both males and females in every study, and so that would be a justifiable exclusion, I would think. If you're talking about quantitative difference, then you're getting a difference in the response of males and females. So, for example, if I look at self-administration behavior, female rats and male rats will nose poke for access to cocaine. And if you increase the response requirement with each subsequent dose, then they will eventually reach a point where they stop and they won't take any more cocaine. That's called the breaking point. If you house females by themselves, they will increase how much they'll work for cocaine and continue to increase over a 10-day period of time. So female rats in isolation work considerably more than males. Females who are paired, here you have a housing condition variable, will work about the same as males, and males aren't affected by housing conditions to the same extent that females are. So you have a quantitative difference, but the behavior looks the same, and is affected differentially by conditions. If you look at distribution, for example, how many females do this versus that, or males, when we look at male and female rats given a choice between cocaine or tasty food pellets, some animals prefer cocaine, some animals prefer pellets. But over a period of time, more and more animals come to prefer the cocaine. In an experiment we published this year, we showed that more females come to prefer cocaine than males do. So about 50% of the females prefer cocaine over pellets. Only about 16% of the males come to do that. But if we look at dopamine release in the nucleus accumbens here, like after the first couple of weeks of self-administration, and here after they've developed their preference, early there's no difference between the ones that prefer cocaine and the ones that don't prefer cocaine. So this is the same. And this is males and females combined, because even though we did enough animals to know what we had 9 males and 9 females early and late, there was no sex difference in the dopamine effect. But after animals came to prefer cocaine, there was a significant decrease in cocaine-induced dopamine in the nucleus accumbens. And so we replicated this effect in these group of animals and showed that there's a population difference in the incidence, but apparently not in the dopamine mechanisms, arguing that studying female rats may be more cost-effective if you want to have a large population of addiction-prone animals, which is something that the addiction research is always, community is always looking for. And then occasionally we find that there are different mechanisms or different brain regions that are mediating the effect. And this is work from Rita Valentino's lab with Elizabeth Van Boxtel and Deborah Bangasser. And what they showed is when they look at corticotropin releasing factor 1 and its response in the hypothalamus, what they see is the intracellular signaling system mediating the response in females to CRF is through a GS signaling system, and in males it's mediated by beta-arrestin. So the same receptor, same coping behavior in the rats, different mechanism mediating the effect. So you can have fundamental differences at that level. And then frequently you have a sex difference in the behavior. For example, here you had, in terms of the implications for women, you have different behaviors that are affected by the stress response in men and women. And you also have a different mechanism. So you can also have greater amphetamine-induced behaviors in females compared to males, and the areas of the striatum we've shown that are activated by amphetamine are different in males and females. So once you have described a sex difference, then one will need to begin to look at the mechanisms and understand what's going on. But first you need to understand what type of sex difference it is that you're looking at. And when you do that, I'm sure you'll hear more about this as the day goes on. Males and females can differ because they secrete different hormones. They may differ because they have different brains or bodies due to the development of developmental influence of genes or hormones. And these two may interact with different hormones acting on brains or bodies that are developmentally different. I want to say something to the biologists in the room when we talk about reporting our data, because I've been collaborating with a woman in history who studies evolution of alcoholism. And for the longest time, she didn't get what I was talking about, because many people have a bias that if the brains are different, if there are sex differences in the brain, then that is causal. That's why everything is causal. And when we talk about humans and when we talk about even rats, there's an interaction between the environment, our culture, and the biology that results in the sex differences that we ultimately are reporting about. And so we need to think a little cautiously about how we report our results so that it's not always blaming the brain for the sex differences or blaming the body for the sex difference when there may be interactions between the brain and the body and the environment in which the people are living. And an example that illustrates this is that after the AMA declared that obesity was a disease, they did this headline appeared in the National Post, just a tabloid. But then the University of Minnesota did a study which found that people who were overweight decided that they didn't have to exercise or diet. If obesity was a disease, then it wasn't their fault, and they could just wait for the pill that would cure them. And I had a similar experience with someone raised a hand at a talk and said, well, if addiction is a disease, then if you use drugs, it's just your fault. It's not your fault, right? You're going to be addicted, you know? As though as soon as you start using drugs, the biological determinism, it's all to the brain, I'm just going to be addicted. So we need to think about how we talk about sex differences and make sure that it's clear that biology is not necessarily destiny. It's simply a part of what we're talking about. So variability in data has many different sources. One of the things that may be a factor are individual differences and sex differences, and these are interacting and need to be thought about as we look at our data. The better we understand the ways that males and females are different in our studies, the better we can construct our experiments. And there will be some experiments where females are more cost-effective than males. Other times that males may be the better model. But we have to study both before we know that. And finally, the mechanisms mediating the development and expression of sex differences are going to advise our science and the translation of that knowledge to the benefit for both men and women. Thank you.
Okay, our next speaker comes to us from the University of Texas System. Patricia Hurd is going to talk about sex in the dish, exploring cellular and molecular approaches.
Thank you so much, and a thank you, good morning to all of you. Thank you to the organizers for the opportunity to be here. I was asked to go a little bit farther into the weeds here, and since this is a session on techniques and methods, et cetera, and so we'll do that to a certain degree. And I think we'll entirely focus on really cellular and molecular approaches rather than the kinds of in vivo things we've looked at earlier. For those of you who know me, I am a stroke researcher, and the reason that I got into sex differences is right on the board in front of you. That contrary to what many of us believed, the disease we call stroke is a very sexually dimorphic disease. If you look at just the incidence and patterns throughout the lifespan between men and women, you'll see that well into the years beyond menopause, men have a higher incidence of stroke than women do. That's not the story, but that is in fact what made us, my laboratories and many of my colleagues, fellows who now are in the area as well, decide if we could understand more about the biology of this and find some better treatments for stroke, which, as many of you may know, we have very little out there. So to focus on just how one might start understanding the effects of sex on a disease like stroke, we did a lot of work in vivo, but I wanted to focus today strictly on in vitro. So for those of you who don't work in this kind of thing, this is a picture of cell culture. On the left, you have primary cultures of neurons growing in the dish. They're typically harvested at particular points that we know from embryonic tissue divide and grow well. And what we do, because we're interested in damage, is we remove the oxygen and glucose from the media of these cells. They go on to die, not at all surprising. We can then stain, count, and study the basis under which these cells do die. This has been used for many, many years. This is a very standard technique. You can grow many kinds of cells from the brain in this particular way. What we took a slightly different tact was, and this was in the early 90s, to say, if we separated the sexes that you grow in the dishes, if you looked at male versus female cells in the dish, what would happen? And this was entirely never done at that time. And the primary reason that we heard it wasn't done was because mice and rats were probably too small to do this. So I want to tell the first story here of about how really we saw that cells respond quite differently to injury. To do that, we had to create these techniques. And the argument from my field at the time was mice and rats, particularly mice pups, were too small to sex. So to be able to counteract this, we had to go back to veterinary manuals, and I think these were from the 1920s. My colleague, who is now here at NIH, Stephanie Murphy, brought this to us and said, there's a real physical way you could see quickly the difference between male and female pups, and you could stratify your cultures and set up parallel culture systems without much difficulty. And when we began to do that, again, was very unknown to do that, so we wanted to validate it. And in fact, you could see this quite readily in the animals from which we set up primary cultures. But to further validate that, we sexed cells. And in fact, as everyone in this audience knows, every cell has a sex. And you can determine that very readily by male cells will show the gene and the protein for the SRY, the testes-forming gene, whereas female cells do not. So we validated all of these cultures in our first presentations to the community and said, this is how you can very quickly and effectively set up cell cultures. There are many technical obstacles that had to be handled. And for the people in this group, many of you I know have set up very innovative techniques. You can appreciate all the little hurdles that you have to get over to be able to make this work well. And in the interest of NIH's very strong emphasis, which I totally support on reproducibility, it was important to publish all the details early about how these culture systems worked. One that was very important was that traditional media that you use for growing these things had steroids in them. So we had to create media that were a bit different. So as many of you can imagine, there were various kinds of things that we had to do to set these up. And some of the early data that we came up with, these are from astrocytes in the brain, is we did exactly what I showed you. You grow them in the dish, you take away their oxygen, you take away their glucose, and you see what their death rate is. And that's what you see in front of you, is the death on the y-axis between male and female cells, primary cultures and astrocytes, and for the first time showed that female cells were relatively resistant to this kind of insult, somewhat correlative way downstream from what I showed you about the difference between men and women in their stroke patterns. We went on to show, and many have done this, is that under conditions of injury, that it wasn't just that there was some innate difference, there are differences, but in fact, you could even look at your particular protein, RNA, gene of interest and show that it might be different in these particular cell cultures. This is, again, data from your astrocyte cultures showing that a very important enzyme is expressed differently in male and female cells. So there are many examples now after injury of molecular signaling partners that are different in male and female brains. So we wanted to try to get a little bit, and I share this with you again just from the orientation of technique development and methods. We wanted not a proof of concept, this is not that strong, but at least a crude beginning to look at why would these cells, male and female, be so different in their ability to tolerate something fundamental, lack of oxygen. And we found that if we went back to papers published by many in the room here of when the rodent brain was masculinized in behavioral terms, and we went in and treated animals in utero very early on with flutamide to be able to block the so-called masculinization of the brain that happens, then derive our cells from those animals, plate them out exactly as I showed you, and test what would happen, we found still female cells were much more robust in their ability to tolerate this lack of oxygen. And in those animals who had been reversed in their masculinization, and I'm saying that with very strong quotes around it, they in fact could look very much like female cells. So the next experiment anyone in the room would do would be to do the opposite. To say, okay, if we go into the brain at this point in time, females be able to produce, to instill testosterone at the key periods of time that they would need, and then repeated the experiment, what would we see? So here again, you see female cells are much more robust in being able to tolerate a very fundamental insult. And if mothers or pups or both were treated with testosterone at key periods of time, you could in fact changed their sensitivity to oxygenation. Again, not a proof of concept, but the beginning, an important set of data to convince my field that this wasn't a sleight of hand, that in fact there was a very strong biological basis for that. I wanted to also make very quickly some comments about mechanism. And Jill's already set that stage very nicely, but I think we possibly could go a little bit more deeply into that because I think that's going to be important for the field in the future. There are some in my field, molecular mechanisms, which are designed, derived, and pulled into under injury paradigms very, very differently in males than females. And I have also studied hundreds of other mechanisms which are not different. And so the most fundamental thing that I always share with people in my own community is that it's so important to look, and once you find that there's no difference, then you go down one path depending on what your interests are. But if you do find those differences, then you are ethically obligated to track those. And let me show you an example of this. This is not from my own work or for my colleagues. This is a paper of Bob Clarks, who was at the time a young neuroscientist and a pediatric intensivist at the University of Pittsburgh. And Bob had heard, as a young person, me talking about this field, and because his interest was in traumatic brain injury, he decided to set up in his laboratory culture systems, and as you can see, this was early on, where he would systematically look at male and female neurons in this case, and look at the traditional kinds of probes we use pharmacologically to study cell death signaling pathways. So what you see on this slide is MTT on the Y, which is the dye indicator we're using for survivability, control cells X and Y, obviously 100% survival, and then what you see are the kinds of pharmacological probes that would be used to test, for example, hydrogen peroxide, to be able to test oxygen injury, to be able to test neurotransmitter injury, etoposide and STS, the kinds of probes that are used in many systems to look for the effects of apoptosis. And what you can see as you look through here is that even in the dish, these cells' abilities to respond to each one of these different probes turned out to be, in many cases, very sex-specific. Others, for example, oxidant injury, and then many years of data subsequent to that, show that, in fact, this isn't something that's very different between male and female neurons. But in pathways having to do with this case, with peroxynitrite, or with neurotransmitter-related injuries, there was absolutely a sensitivity of males. And you would then design your drugs using these kind of data very differently than you would for males than you would for females. So this was one of the first studies that actually showed very commonly used techniques and how different they could be when you applied them to sex-stratified culture systems. And I wanted to summarize, instead of going through all the data, how really significant this could be. After that paper was published, and I have to say that this young neurointensivist never went back to studying anything about sex. Published this one paper, very important, that was it. That was something that had caught his interest, and that was it. But there were a number of us that continued to work in the area. And in fact, the data here, that I'm not going to go through all the details, I just want to show you the pathway, really belongs to two fellows, Mingyue Liu and Louise McCullough, who were both fellows in my lab at the time. And they went through this whole pathway, which is put in most medical school textbooks, of how one injures neurons. One begins with ischemia. These cells are electrically active, they depolarize, they release neurotransmitters, they engage an enzyme called nitric oxide synthase, and there's a whole series of events that occur down here. At last count, there was something close to, I think it was 17,000 papers published on this entire pathway. And in fact, I cannot tell you how many drugs were developed, several of which did in fact go to clinical trials and failed, all based on this pathway involving these molecular players. And this is one of the most striking pathways that I know of, which diverts at this point. This is absolutely a male-dominated pathway, right down to where you have mitochondrial injury and changes that are happening in the cytosol. And for the females at this point in time, Female cells are totally indifferent. Larry very kindly showed earlier the paper that we published in 2005 showing that even in vivo, if you gave inhibitors to be able to block this part, females not only didn't respond well, they got worse. So this is a fundamental thing of where the molecular systems diverge, and I think Jill showed one very beautifully in a very different area, that this entire thing is wired differently. And whether we should develop sex-specific drugs or not is a question I don't know that we'll talk about today. I did want to comment on the Lazaroid story, and I was a Lazaroid investigator, so actually the piece of that was not even that it was different mechanistically, it was about pharmacokinetics, that the amount of drug that women needed was different. So it's a complex kind of thing that we have to think about before we make decisions of if we would develop sex-specific drugs or not. I'm not proselytizing for or against it, but it is a complicated one. So let me stop there with just the concept of, first of all, there are techniques that have to be developed to be able to handle things like sex-stratified cell culture systems. There are molecular approaches where you can show that even at very fundamental kinds of levels that male and female cells do respond differently. And what I showed you today has been, for me and for many of my colleagues, a very powerful argument in the community where people say, I'm very interested in mechanism. I am not interested in sex differences. So if one shows mechanistic pathways which are sex-specific, sometimes that is one of the best arguments that we can offer. Okay, let me stop there and take any questions that you may have.
Surely somebody has a question. Ah, good.
Beg does.
So why haven't they figured out the pathway in the female?
Well, actually, there is the way that happens is Louise McCullough's work. And what happens is that instead of taking the nitric oxide, peroxynitrate pathway, many of those cells are shunted into apoptosis using caspases. And so we do know something about that pathway. But
and do you think that that's hormonally directed or sex chromosome directed?
Not tested in either way, except to give you sort of the epiphenomenon that if you remove sex steroids from the animals, so we're now talking in vivo, you will not see that, in fact, that determination of their sensitivity is different. So at the very simplest level, we know that it's not mediated through estrogen, but that is, again, very high-level response to it. And I don't know of anyone who's looked at it chromosomally. Yeah. Art?
Louise has done a recent stroke study and fails to find a sex chromosome effect in the sex differences in stroke. And so at least at that level, there's no evidence of a sex chromosome effect, so it's likely hormonal. That's it. It's stroking animals as opposed to cell cultures.
Thank you. Hey, Virginia.
Thanks very much for that. I think we'll have our work cut out for us more so in the cellular molecular community than we do maybe on the individual scientists who work with whole animals, because those differences may be more obvious. I was at an NIH conference a couple of weeks ago in the Sanctuary building talking about neurovascular interactions in stroke, where investigators stood up and said he's never seen sex differences in cells in a dish. The whole concept is witchcraft, and why pay attention to it? So I think the point that you make, and it's very important in terms of techniques that need to be developed to understand the differences if you have cells from one sex to another, the culture environment may modify some of those differences that may exist naturally. And I'm not sure how to get those concepts to molecular biologists to understand them. I think it's a little bit easier to work with whole individuals who work with on a more whole animal level than the molecular level. And so I would appreciate your comments on how you might reinforce those comments or concepts to the molecular community.
A terrific question. I also wanted to put the slide up real quickly, which you'll be able to immediately see. These are endothelial cell cultures from microvessels in brain, segmented out exactly as I've shown you earlier. And as you can see, the responses of female cells are quite different than males in the dish. So there are very much neurovascular kinds of differences. What I have found to be extremely effective when I've talked to colleagues working at the cellular and molecular levels is we have to get in the weeds. It's not a high-level discussion. And I think it typically has to do with reproducibility as well, because most of us that work in these systems know that you have to be extremely careful to be able to reproduce these results because the conditions are so artificial in that regard. And so the most effective thing for me, Virginia, has been really getting into the how do you do the cultures, what does it mean, what difference is it? And then the other thing that's worked extremely well as an argument for me has been about signal-to-noise ratio. Nothing is harder for a really strong scientist to have your pet hypothesis go awry, where you put it out there, you do the key experiments, and they all come up showing that your hypothesis is wrong, or even worse, that you have no difference. So taking that tact where you can show that there's a very strong difference when you sex your cells, it allows someone to pursue their pet hypothesis and show that they were right from the very beginning when they sex their cells. And that has happened a number of times. And so there's another thing which I found to be very, very effective to talking to really strong molecular and cellular scientists, because they have very important hypotheses they haven't been able to demonstrate simply because they were working with mixed sex cells.
Following up on that, it's absolutely critical then that the scientist who does that makes clear in the paper that this was in one sex and not the other, and that's generally where it falls apart still.
Well, I think we had some very good discussion about that earlier, about how as a community we have to set some standards for how we report the conditions and the outcomes along the sex specificity. And I think we also have to look at that in, again, a much more general way, that we're well aware that most publication sources don't allow you to tell anything about your methods. It's just been part of the community for a long time, and it's probably time for us to take a look at that as well under the banner of reproducibility, not just sex steroids. But your point's very, very well taken. Yeah, Farida.
I just wanted to actually ask the question that Virginia raised, and that is, besides the importance of looking at the sex of the cells, it's also important, if you're studying, that it be congruent to the model that you're studying. So very often in the case of adult diseases like stroke, hypertension, etc., you're only answering a part of the question if you look at fetal and neonatal cells. And if you take adult cells, which is really the cell type you want to be studying, you start to notice some of these. So we've been looking at adult and aging cells, and when you look at aging cells, you don't see the sex difference, just like you don't see it with the strokes in the aging animals. And in the younger cells, you see the sex differences. So the cell model is very powerful if you try to replicate as many aspects of your disease as you possibly can.
Well taken, and there are some technical limitations on that. For example, the data that I showed you in brain endothelium, those are taken from adults. Very difficult to culture. Whereas for neurons, if you want to do those in adults, I wouldn't believe the data that you would get, neither would you. Yes, sorry, question?
So you made an important comment, very simple. If there are no differences, we should report that there are no differences. If there are differences, we have an ethical responsibility to study the differences. So I think one of the purposes of today is to advise the NIH, how do we specifically state there are no differences? And if that is stated clearly, will that be enough as an exemption for your grant application?
Well, perhaps I can answer from someone who has been, fortunately, extremely well funded by the NIH for a long time. And what I have done in every single application and every paper that I've done in which I only publish one sex is that I always put the data in there, and sometimes in a paper with just a few simple lines to say, we studied this in both sexes. We report the data as such. Because there were no differences, we then pursued the next steps in a single sex or a single sex of cells, or however we chose to do it, with that rationale. And I have to admit that in my years, I have never had pushback from anything I've ever published or any of my grants along those lines. The reason I think it's important to say that is that I hope one of the issues we talk about today is that for young people particularly to be doing well in this field and to deal with the costs of experiments, as we are well aware of, we have to be very clear about what we're asking them to do. And I think that when you've done the work and shown there's no difference, then there's no reason to keep pursuing it. And we as a community need to be able to say how, as you were saying, very clearly make that point. The ethical responsibility, you have to remember, my comment comes from someone who has always been in clinical departments and has very strong connections to clinical science. And I think it's very, very difficult, and I have designed clinical trials knowing that I don't have any preclinical data in women. It's extremely difficult to do. So I would argue, hopefully to stimulate vigorous discussion here, that it is a responsibility that we have to have if we have any interest in our research informing clinical and translational research. Yep. Yes?
So I just want to say that I agree with you that we have to be well prepared to say there is no difference in a particular endpoint at a particular time under a particular circumstance. What we have to be very guarded against is whitewashing that the entire phenomenon has no sex difference. And the way we protect against that is going forward, it can't be, okay, there is no sex difference, so now I can go back to studying only males. If there is no sex difference, there is no reason for you to study only one sex. And all of your future experiments should include both sexes. And that would then allow you to be able to detect if a sex difference appears later. It doesn't mean you have to double your N, because you've just told me that there's no sex difference. So you can have your N of 10 be 5 males, 5 females, and in fact you'll save money because you're not throwing away half the litter, et cetera. And the big concern is everybody's going to say, nope, I checked that box, no sex difference, I go back to my old ways and do only males, and then we have not achieved the goal of equal representation.
I think those are very good points. That's obviously not what I was suggesting, but it's a very good point. Yeah. Virginia, and then we quit.
Can we...
That is where you would run into problems with these drug developments, that you're looking at the endpoint and biologically, but the mechanistic pathways to get there are different. So I think we have to be very, very careful when we declare that there's no difference, because it's what difference are we actually looking for, and how do you get there?
Okay, thanks to everyone for those questions. I'm sure we'll be continuing them as we move to the end of this session. Our next and last speaker of this session is Wai-Yung Chen from Texas A&M. And he's going to tell us about enhancement of research findings, the significance of well-designed experiments, and I'm sure that we'll be hearing a little bit about statistical design.
Well, first, thank you for the organizer for having me to participate in this workshop. And what I'm going to talk about today is a little bit different. I'm going to talk about the fundamental regarding the experimental design and maybe the ways to include sex into the experimental design. So a few years ago, our department and actually has a journal club, and so we actually talk about this article. So if you look at this from the New Yorker, and the title is "The Truth Wears Off." So this article starts talking about why their second generations of antipsychotic drugs, actually their effect is shrinking. So if you look at this, actually the issue probably relies on this very small subtitle. Is there something wrong with the scientific method? Well, just a years ago, another publisher in The Economist right here, and it's very frank and just talk about in the title, Unreliable Research, Troubles at Lab. So this article also started talking about the research published, and I think it's in Germany. So they're talking about trying to replicate 67 publications, mostly in I think oncology, women's health, and cardiovascular. So among those 67 studies, they can only replicate 21 to 33% of the outcome. So there is a really probably fundamental issues regarding the reproducibility issues here. So obviously everybody know about this article, and then Dr. Tabak talked about this a little bit earlier today. So within this article, actually, there are very few important points. One it talks about poor training in terms of the experimental design. And the second issue is lack of disclosure of the basic element of the methods. And then the other one is the politics, okay, the political issues and attitudes from the funding agencies and institutions and publishers. So from the institution standpoint, many of you probably realize that saying, well, if you want to get promoted to get the tenure, they want to count how many publications or what's the impact factors of your publication. That may impact in terms of the research outcome as well for the publisher. And I think that we just talk about the issues regarding if there's no differences between the male and female. The idea is that the publisher should consider publish anything, even it's what negative data. So I think those are important for them to change their attitude. Even though right now, I think starting the last year and the many leading journals in science, for example, Nature and Science, they have changed and they have to raise their standard and they're trying to include more information into the method. So hopefully that people will be able to follow those methods and will be able to reproduce those data as published. So among those three things right here, and obviously what I'm going to focus on about today is probably talk about the training in terms of the experimental design and in terms of the disclosure of the information within your study. So this is actually a publication. It's a survey published in 2009 in the +1 here. So I just take one table from their surveys right here, talk about the numbers of study reporting sex of the animals. So if you look at these 271 publications here, and there's mice, primates, and rats, and you can see there is still a substantial number of the study didn't even report the sex of their subjects. So fortunately, we have a new guideline, recommendations right here. So not only you need to report the sex of your subjects right now, it's recommended that you actually to include both male and female in your study here. So how are we going to include sex in our experimental design here? So if sex is a factor of your research interest, so obviously it's very straightforward. You can just include sex as one of your independent variable. What I'm talking about independent variable here is talking about the factors that you actually manipulate. Okay, so the thing is, if sex factors is not of your research interest, I would still recommend that you will include both male and the female in your study here. We're not talking about this table here, okay? I think that many of you are familiar with the type 1 and type 2 error in your analysis here, because people can argue saying that if I include both male and female in my study, it may increase subject variability. The thing is, we're not talking about because if you increase your subject variability, obviously you're going to increase what we call the type 2 error, which means that the null hypothesis actually is wrong, but you accept it. So what that means is we are not talking about from this stats standpoint. We're talking about whether you are able to generalize your data into a real world. Because if you only use one specific sex right here, so the data can only generalize to that particular sex. But now even though you're not interested, or even maybe there's no differences, I think that it's important to include both sexes in your study, because now your data can be generalized into both genders and both sexes and in the real world here, okay? So how are we going to analyze the data if sex is one of the independent variable here? Well, if sex is the only variable, that's very straightforward, because now you only have male and a female. So it's very easy. You can do all the simple t-tests, okay? Or you can use the Mann-Whitney U we're talking about here. We're talking about two groups comparisons right here. So you have to be aware that because a lot of the analysis, if you see for the publication here, and sometimes they don't really care about where the data coming from, because if your data coming from what we call the ratio or the interval, which means that each individual unit between those units are equal, and then you can use the t-test. But the thing is, a lot of times your data actually come from a different scales of measurement. For example, your data come from the ordinal, which means like your rating or your ranking of depression levels, or we'll follow those football rankings every week. So number 1, number 2, because the difference between those 1 and 2, 2 and 3 are not equal. So those data cannot be simply just analyzed by the t-test. There are specific tests designed for those dependent variable what you measure. A lot of times the researchers don't even pay attention to that, and the data can be supposed maybe not going to be significant, and because they're using the wrong analysis, and the data turn out to be significant. So if sex is one of the 2 independent variable, well, that's what Dr. Tabak was talking about a little bit earlier. Factorial design is a good design, because now you can, using what we call the 2-way analysis of variance right here, all depend upon what the other variable is, with what we call the within subject design, and then you will have to use the mixed ANOVA right here. So if I give you a very simple factorial design, maybe 2 by 2, so you have male and a female, and then you have maybe another drug treatment, drugs or no drug. So basically right now you have 4 groups right there. So male, drug, male, no drug, female drugs, and female, no drugs. That's a very simple factorial design. But the thing is, if you go back to the literature, you probably will see a lot of the researchers will use a multiple t-test to analyze that factorial design. Because that's easy to do, pairwise comparison. The thing is, not knowing when you do multiple t-tests is going to inflate your alpha, which means you increase your chances of getting the positive result. In reality, there's no such positive finding. So using the right analysis, I think it's very important. So even though you do have a perfect 2 by 2 factorial design, how you analyze data is critically important. And then don't just take the easy way by doing multiple t-tests, or you're using a 1-way ANOVA. You have 4 groups right there, and then there's a significant F, and then you do the multiple post hoc comparisons. Those also will lead to the increase in terms of your alpha level there. So if your sex is one of the multiple, more than 3 independent variables right there, and my recommendation is change your design, rethink. Because why? If you apply 4-way ANOVA, most likely you're going to get 4 manufactured of those each individual factors. You're going to get 6 2 factors interaction. You're going to get 4 3-way interactions. You're going to have 1 4-way interaction. And I can guarantee you that there's no way you can interpret those interactions, if they don't go into different directions. So if you have to design a very complex design and then rethink about what's the best approach, because you also have to consider whether your analysis, whether the outcome, you can really interpret those data. So what's the advantage of including sex as one of your independent variable? We touch on this a little bit later, because a little bit earlier. Because why? The factorial design actually will increase your power of your study. Because why? You are increasing your sample size. So if you look at this very simple 2 by 2 design right here, you have 1 factor is the sex, male and the female. And a dose 1, and maybe another group is a control here. So within these 4 groups here, each group you have 10 subjects, for example. But the thing is, if you're using the factorial design and using the appropriate analysis, so the analysis for your the dose 1 and dose 2, instead of having 10 subjects, now you actually have 20 subjects. So you double your sample size. So if you look at your sex factors right here, so your male not only have 10 animals per group, you have 20. So that will significantly increase your sample size in your analysis. So which means that it's a good idea to include the sex as one of the factor, and then changing from a simple design into a factorial design. So the other thing is the factorial design also allow you to study the interactions between or among factors here. So if I just give you a very simple designs right here, so you have a design wanting to study the effects of the drug on the locomotive activity, and then your outcome is that your dependent variable is to measure the distance traveled within the open field. So that's a very simple 2x3 designs right here, 2 factors. There's a sex factor here, male and the female here, and then you have those 1, 2, and another control here. So if you look at the outcome, this is a dependent measure already, the outcome right here. So if you look at these data crossing all 3 treatment groups right here, between the males and the female, if you add it all together and divide it by the 3 groups, you're going to get what? The equal means right there. So what that means is your conclusion may be, if you don't using a correct factorial and the two-way ANOVA to analyze the data, you might say, well, there's no differences between what? The male and the female. The fact is what? There is interactions actually between the gender, between the sex and what? And the doses that you're using right here. For example, if you look at the dose one here, it will increase the locomotive activity, but this will only observed in female when you are giving a different dose. So the factorial design will allow you actually study the interactions. So this is a very powerful design. So now the question is, so what is the adequate, the sample size for experiment? I think this is an extremely tough question to answer, okay? Because the sample size is not dependent upon just one single factor. So they're dependent on the 3 here. One is, what is your expected power? Well, all of us probably will expect that our studies, the power is probably what? 0.08. That's what probably the most everybody will accept the level here, 0.08. So what about the second issue here? What's the sample size here? It's dependent upon what? Your effect size here. Okay, so you need to get those information before you actually can calculate how many subjects actually you need for your study. So how are you going to estimate in terms of your effect size right here? Well, most likely you're going to using your prior research experience there. So all the published data, either yours or your colleagues, or you can do some preliminary studies and you can get those data. Well, sometimes you can probably expect in terms of what's your effect size of your study. For example, if you want to inject alcohol into an animal, and I would assume that you will see a huge increase in terms of their blood alcohol concentration. So what that means, you expect the effect size is going to be large. The thing is, if you inject the alcohol and try to see whether the alcohol will change the brain size, well, that probably the effect size could be a little bit smaller than what you expect to see the alcohol within the blood. In that case, you probably need to introduce a lot more subjects into your study. So the other thing is you also have to consider your significance level right here. And most of us expect the 0.01 or 0.05 as standard for analysis to become significant. So I just give you a very quick tables right here. So this is the sample size as a function of the power, effect size, and the significance right here. So here, the red color, that's if your alpha is a 0.05, and then the blue is if you're a little bit more stringent and you want to go to the 0.01. So those are the effect size, just very grossly divided into small, medium, and then large right here. So you can see now, if your effect size is small, and then you expect to just normal 0.05, and then you need to and also you expect your the power is 0.08. So you are talking about a significant, huge number of subjects right here. So most of us probably all expect our effect size is not that small. And so we'll always expect our effect size a little bit large right here, because that's what you believe. That's what you want to see. So which means still there's a substantial number subject is needed actually for you to really say that this data is going to generate a high power, and also the high power, which means that people were likely to be able to replicate your data. So this is publications in just last year from the Nature Review Neuroscience right here. So they look at the 48 published the paper, and then they calculate in terms of their power based on the sample size they report. So this is in the neuroscience here. If you look at these, so the x-axis is right here is the power, and then what? The y-axis is the number of the publication here. So if you look at this figure here, among those 48 publications, and then many of those publications, what? Their power is below the 0.8. And then you'd be surprised, actually, there are 15 publications, their power is less than 0.1 because what? The sample size is not sufficient to actually get a power. So how to increase the power of the study? Well, obviously, we were reducing the confounding variable. When you're doing a study, you want to make sure that your treatment stands out. Because if you understand about the ANOVA here, so we're talking about variances here. So you want to make sure that your treatment variance is on the It's good, but the thing is you want to because that's what you couldn't avoid it. That's what you're measuring. But the thing is you want to make sure all the other variables, the variance decreases so that you can increase your F value there. So now by decreasing the variance comes from the confounding variable, and obviously that will increase the likelihood you are going to get a significant outcome there. So what confounding variable I'm talking about here? Okay, I think some of we have a touch on this a little bit earlier. For example, the equipment you use, whether you calibrate or not before every single exam, every single test, or the chemicals you use. Are you using the same grades? Who's making the solutions? Are you making the solution follow exactly the same protocol? The testing environment, or what? Sometimes even the housing environment, whether you house the same treatment animals always on the top shelf of the rack, or all the other groups on the bottom. So those may have the impact in terms of your outcome there. The other issue you can control is experimenter here. We also talk about whether someone is very experienced or is very seasoned compared with someone afraid of the rats, grab the rats really tightly, and then causing the stress of the animal, and then it may significantly affect your outcome. The other thing is you may even have to consider whether the male or the female experimenters in your lab. There's this new publication just this year in the Nature Methods talking about the presence of the male experimenter or the sense of the male actually induce the stress induced related analgesia. So which means that if you want to study pain study there, if you're using the experimenter as the male or experimenter the female, your outcome is different because at the presence of the male experimenter, or it can just have someone wearing the male clothing, maybe not bathing for days, and they still have the sense of the male, okay? And the rest will not respond well because they have stress-induced analgesia right there. So the other issue is probably to reduce the subject variability here, okay? So I will probably jump to the very last one right here, okay? Because if you can introduce your design using the within subject design, okay, not only you can decrease the number of the subjects, at the same time you can also decrease the subject variability because now you have the same subject exposed to all the level of your the other variables there. So I want to just last slides right here is so what can we do as a faculty mentor the researchers on this how to enhance in terms of the research here. So I think education is still the one thing right here. So, for example, at our institution right now, we do have a course. As a matter of fact, that this course was developed when Dr. Susan Meyer, when she was during her tenure in Texas, and we created this course right here. We're not teaching stats, okay? We have to make sure that we're clear about that, okay? Because we're teaching here is in terms of the experimental design and how to design a study and how to apply apply the correct analysis into their studies there. So I think that we all bear that responsibility, probably be able to teach the newer generation, the younger generation, the researchers to be able to understand the importance of the experimental design here. I'm sorry, maybe I went a little bit longer, so I will stop right here. Okay, thank you.
Okay, if we have one question or so for Dr. Shen, but everybody else, ladies, could you please come up to the table? Because we have a few minutes to have some crosstalk.
Perhaps just one question for the last speaker before the general discussion, and it's more along the lines of being an apologist for basic scientists per se, based on one of your points earlier regarding the Bayer Healthcare study, suggesting that one of the issues of reproducibility is that there's a lack of translatability of bench science into pharma. And that was picked up by The Economist, as you mentioned, and it was published by Bayer Healthcare out of Germany. The authors of that paper were the Vice President for Research and two other leaders at Bayer Healthcare. And I just want to note for the record that, in fact, their methods were to send out a survey to the leaders of the various programs at Bayer Healthcare to ask, is there reproducibility of the science that we license in? So the methods were not ordinary. The survey has to be acquired by emailing the first author, and that has not been forthcoming to get that method. And I think until there's really a rigorous assessment of whether or not that reproducibility exists, the conflict of interest for industry to suggest that the work that is being licensed from the basic science cannot be reproduced is only self-serving. And I think that's an important point to make for the record. And you indicated that some of the issue is really about, on the academic side, the need to publish or perish, the need for tenure, the need to be in high-impact journals. And certainly, as the very elegant article by Collins and Tabak pointed out, this certainly is one of the issues on our side. But I think we should avoid a blanket statement that says that we are not reproducing our own data on as broad a scale as possible. Issues like sex inclusion, issues like statistical methods, these are improvements that have to continue to be made in the scientific pipeline. But that one paper itself is so deeply flawed that I hope that as a scientific community we don't rely on that as our mea culpa. We have a lot of mea culpa that we must take on. But that one paper from Bayer Healthcare is not the smoking gun. There are other reasons that we are here. And so I just wanted to correct that kind of perhaps, or put that comment into the record for today's deliberation.
Well, thank you. And the thing is, within that
one that looks like a mouth.
There you go.
There you go.
Okay, thank you. I agree 100%. Saying the thing is that when they suggest in terms of the data cannot be reproduced, they're never blaming. So in general, they are not blaming in terms of their any kind of suggesting any flaw in terms of the research. So, and I think for all those papers, for example, like the New Yorkers or the Economist, they cite a lot of the research. Some of them, they are basic science research too. And obviously, the driving force behind that, you're 100% correct, because a pharmaceutical company actually driven a lot of those discussions right there. The thing is not related to in terms of whether anybody is committed any misconduct in their research so that the data cannot be reproduced. Rather, it's because maybe a lot of the information has not been disclosed. So a lot of things, if we cannot really follow exactly what you're doing, and I cannot replicate the data, just like if there's no indication in terms of the sex, how do I know which one I should use? Both genders, both sex, or just one? So it's really not reflecting anything regarding the academia, the research. And even though sometimes for a researcher, if you wanted to replicate your data, sometimes it's hard. So there's a very well-known alcohol researcher, for example, and then he tried to replicate his own data at 3 different locations, 2 countries, Canada and the United States, at 3 different locations. He will be there, the same experimenter, the same thing. The thing is, he cannot replicate his data at 3 different locations. So there are things that maybe we still need to be aware, and that's why I will probably also recommend that all researchers probably need to maybe pay attention to a lot of the things that happen in other fields. For example, I will never know if I don't read those papers regarding there's male and the female experimenters. It could be what resulted in a different outcome. And the thing is, maybe back into 1990, early 1990s, okay, if there's no publication talk about in terms of the literary fact, if you study the prenatal studies, and then you cannot just, I just using one female and to get all the pups, I use all the pups in all the studies. But there's a little fact there. So those are the confounding variables I'm talking about. So, but I appreciate that you point that out.
Are there any questions that you'd like to address either to a specific panelist or to the whole panel?
Good morning. I am absolutely not a laboratory preclinical researcher, so I have learned a lot today. I love the comment about you got to make sure what your medium is made out of. I mean, these are really very enlightening for me. What I have heard this morning is basically about neuroscientific research or behavioral research based on sort of our neurology or our, what we're trying to do with our brain in addiction, for example. Are there other examples in cardiology, pulmonology? I don't want to go to endocrinology because that is really a sex-specific thing, but maybe not in adrenal cortisones or something, adrenal stuff. So do we have, or is neuroscience the strongest area of interest in preclinical research?
Well, I just wanted to make a comment that when you look ahead for the day of the program that we have, you're going to hear from people in all the different areas, and you're going to hear the same theme of the sex differences and how really profound they are. So I didn't want to answer the question except just say it happens that some of the people that you've heard from this morning are either on the vascular side, on the neuro side, but I think we're going to hear, and looking around the audience, there's some very strong people here in heart and pulmonary, et cetera. So I think we're going to hear a lot about that.
Any other questions? If not, what I'd like to do is ask each of our panelists in turn to think about what it is that the study of sex differences in either preclinical or translational research, what kinds of scientific opportunities are there to learn some new things that might be much more applicable or make our clinical work and our epidemiology work and so forth have much more of an impact on the population?
Well, I'll just say as someone who's had an opportunity to do drug development and to take those from the bench to full clinical trials, that I think one of the opportunities is to really shape where drug development, device development, things like this are going to go. That is the strength of our community preclinically. And by increasing, not only dealing with the issue of reproducibility, which I think is certainly an umbrella one over all of this, but by strengthening our science, which is what we heard about very early on, that we are going to have huge implications for the development of healthcare that's going to go forward in our country. And I think that's something we hear about every day. So without getting too high level, I think that's one of the hugest opportunities, is to really better shape the expensive processes that we go through for drug and therapy development and make it much better informed.
So I agree. I think the example of the sex differences in the mechanisms mediating the response to CRF is just the tip of the iceberg in terms of understanding the mechanisms through which sex differences in the response to all types of neuronal signaling is happening. And I believe that as we start getting beyond just describing the sex differences that exist and understand how they come to be affected in the brain and other areas of the body, that we will have better targets for drug development that will be appropriate for both males and females.
Yeah, I think this, my comment probably is not specific just to the preclinical studies or the translational studies. I think including both male and female in all fields, I think it's important. Not because from this statistical standpoint, it's actually from what? From the how you interpret the data standpoint. So because in our society now it consists of what, both male and the female. So in order for your data would be what? Be able to generalize it to the population, you need to include both male and female. And then, so even though if you're not interested in that the particular factor, but the thing is, if you do have to increase both male and female in your study, why not just separate that as well, one factor, and then be able to take advantage of all the other experimental design, and then what? So that your data will be what? More reproducible, and then what will be likely to be able to generate to the population.
Okay, I guess we have a couple minutes or not. We have some more questions. This is good.
So I wanted to ask Dr. Chen about the issue of sample size. And so you gave a sample size of N of 10 per group for males and females. I think most of us would kind of faint at the notion of having to have a sample size that large. So say you did your power analysis based on the published literature in males and your pilot data in males, and your power analysis said that your N needed to be 8. What should you do in terms of including males and females for an N to go forward? Can you do 4 males and 4 females, or do you have to do 8 males and 8 females?
The guideline is if the sample size calculation, that's based on what? The individual groups. So in that case, in any groups, you should have what? At least 8 samples, 8 subjects within the design there. So, so I would say I'll probably go for what? The 8 per group. 8 male and what? 8 female. So.
Right. That's a huge problem. If that's what we're recommending, we're going to get huge pushback.
The thing is, but the thing we all know, the sample size is critically important to determine the power. But if you don't have enough sample size, that's the consequences, that the reproducibility issues there. So I think that is hopefully through the education will be able to convince the people that this is the sample size that you need in order to get a meaningful outcome. Otherwise, you can invest all the money at the end, you will not be able to generalize to the population. But the thing is, for all the research, you also have to realize one thing, that is that if your hypothesis is wrong, which means that no hypothesis is true, it doesn't matter. You can put 1,000 there. It's not going to be have an outcome. So people trying to think that if I keep putting more animals there, I'm going to get an outcome. Well, if your hypothesis is if your no hypothesis is correct, and it doesn't really matter how many subjects you put in there, the outcome will not become significant. So.
I just wanted to add to that. I think it's also very field dependent. Some fields might not think that large sample sizes are important. I know in my own field, if you tried to publish data in animals in less than a group of 10, you probably couldn't get it published anywhere in a good journal. Not to say that that's good or bad, it's just that's the field's feeling about it. And many would ask for power calculations to show that, in fact, you could do something less, and they might not work out that way. So I think some of it's very field dependent, too, of where our areas are and what our fields demand of us. Sorry, I didn't mean to
Okay, please.
I just want to congratulate the organizers for having the foresight to bring on board a statistician, because I think this is really important. I think one of the take-home messages is that investigators really need to engage a statistician early and often. So I think that's very important.
So accepting that sex might be an important factor, is it not true? First of all, you can't figure out, you can't do a power analysis unless you actually have some data. If you were going to run 10 animals, you're looking at it, let's say, a drug effect. You have 10 males with a drug, 10 males with a placebo, and you're interested in this drug effect, and if you wanted to say, okay, is it also the same in females? Let's say you run 5 males and 5 females in the drug and placebo groups. That would give you the preliminary data, at least, to estimate, to do a power calculation of how many males and females you would need to see if there's a sex difference. Whereas if you can also lump the males and females together to see if there's a drug effect. Okay, if the males and females aren't very different, you can lump them together and see if there's a drug effect without starting out by saying, okay, instead of running 10 males and 20 animals, one in the drug group and one in the placebo, now I got to run 40, male and female drug, male and female placebo. There isn't a way into studying the sex differences that doesn't involve a priori a doubling of N from the very beginning.
Last question.
I think if there is, for example, there is a sex differences in your prior studies there, and then so when you start to incorporate both sex into your study, and in order to get to a significant power for that, I think you still have to go by with what is the highest number that you needed for your experimental design. Because if you don't, you're running the cost of maybe not be able to reproduce your data by using the fewer number of the subjects, maybe from one particular one particular sex there. So
you're saying I need to start with 40, not 20. That's your answer.
If you ask my recommendation, I would say yes.
I would agree. But isn't there a difference between the effect size of the drug and the effect size of the sex difference?
Yeah, right.
They're not necessarily the same.
Right.
There could be a huge effect size of the sex difference and a small effect size of the drug, and you would detect the sex difference without doubling the N.
But the question now is whether you are interested, if both factors are important to you, and I think that I know it's probably as expensive as a lot of the resources, and then the issue to me is still whether that outcome is going to become a significant, going to be meaningful or not. But I think that's what I say. Well, when you have to get involved in terms of the sample size, that it's a very difficult issue to answer, and all the 3 factors you need to consider. And sometimes maybe you can. Well, I know the field right now is always considered 0.05 is the gold standard, but you can change your alpha too if you need to. Okay, so you just have to be aware those information has been reported. Okay, so.
Okay, we have one last question back here.
Yeah, this is just a comment. I think statistics is a tool just like any other scientific thing, and we should not let the statistics drive our science. I think we're so used to thinking in terms of mice and rats, we forget that there are other animal models which using larger N, like sheep for prenatal studies, or pigs, or even dogs or primates, where those large N's are just unattainable and unrealistic, period. And when I think back to all the wonderful data that we used to obtain from these larger animals, which have driven most of what's in the textbooks, it wasn't on N's of 40 or whatever. They were on N's of 6 or 2 or 3, where the effect size was very large. We didn't need to do it. The other thing, what was I going to say? Oh, if we already have a large body of information and data on one sex, it seems to be reasonable to fill that data gap to allow experiments on the sex where the data are missing without doing a direct comparison. And if that and if a comparison with the data that's published looks like there's going to be a significant sex difference that needs to be explored further, then we can talk about how to design the experiments which include both sets of animals. To start out saying that all new experiments going forward have to include both sexes is going to bring the world to a stop. And the scientific community is not going to embrace it to actually progress the science to the next level. I think we have to think about what we need to fill, how we're going to do it, and what is realistic without the reliance so much on rodent models. And I think that has also precluded advancements because we are stuck thinking that the mouse is the perfect animal to study human physiology, which I will push back on.
Well, on that note, we'll take a break until 11 when our next session starts. I guess I didn't look around. There must be a cafeteria over here where you can get some.
Yes, there's a Starbucks and a
coffee.
cafeteria. And for those questions that are still remaining, we have a box outside where you can just write it down on a card for our call of action discussion. Thank you. At this time, Dr. Teresa Woodruff will be coming up. Thank you.
Good morning. My name is Teresa Woodruff, and I'm the director of the Women's Health Research Institute at Northwestern University, and it's my pleasure to be the moderator/provocateur for our session 2 of today's agenda. And the topic that we're going to be considering over the next several minutes is what is the impact of including or not including sex as a basic biological variable. And I think we will have a great dialogue over the kinds of diseases and conditions that are affected by sex and how the inclusion of sex or the absence of sex can actually penalize the research pipeline along the way. We've heard about this in terms of reproducibility, but I think there's a variety of ways that the absence of sex can impact our understanding of health and the treatment of disease. And we'll hear about that from our 2 distinguished speakers. I was thinking as I was listening to the prior several talks about the last time there was a really monumental change in science, and that occurred in 1623, I believe, which is, we have historians in the room, so I fear actually my memory on this, but I believe Francis Bacon in about 1623 published Novum Organum. And this was at a time when there was a great deal of heterogeneity in the way science was being done. And in fact, the 5 senses were largely driving how science was being observed and then ultimately being reported. And the development of the microscope was one of the key features, that new technology that drove a new level of thinking about how science should actually be accomplished. And I'll just remind you that in Novum Organum, the scientific method was laid out, the concept of the hypothesis was described, and the first principle of the scientific method, this new concept of the scientific method, was that the observed fact needed to be repeatable, that you needed to be able to repeat that observation over and over. That is the foundation, the bedrock upon which all of science is based. The second is that it needs to be measurable on a global scale. And this is what I was thinking about with the alcohol discussion, where the researcher went around the globe trying to repeat his own work. But the second principle of the scientific method is that it must be repeatable that it must be measurable on a global scale, that it must be able to be done in other laboratories and with other methods, looking in through a prism on that particular mechanism, from cells to animals to primates to humans. The third is that there must be consilience, that as different groups begin to learn something new about the biology of a particular organ system or a particular fundamental fact, that the ideas and the threads that start coming in to the scientific method begin to make sense, that they become organized in a key way. And you'll remember in 1963, Thomas Kuhn reported on the paradigm. And in that book, of course, he describes the paradigm as a word in 43 different ways. But the principle was that sometimes around times of paradigm shifts come a lot of discourse and discord, in fact, within the scientific community. And it's as the scientific community comes together around fundamental truths that then there is now a new paradigm shift and the scientific field can move forward. And this concept of consilience that was described by Francis Bacon really encapsulates that Kuhnian idea of paradigm shift, and something that I think maybe we're feeling in the environment right now as we're just on the precipice of really making fundamental new discoveries as we all come together and recognize rightly that our ability to reproduce things isn't quite there, and perhaps sex is one of those big variables that we're missing in the equation, our ability to report it and then for others to be able to measure it. The final thing that Bacon said is that the scientific method must be therefore heuristic, or it is implicitly heuristic. And that is that as I come to a conclusion of my study, if that study is really correct, there are open doors that come immediately thereafter, that once we get to a conclusion about a hypothesis, it always opens new areas for discovery. That concept of heuristic is why when my mother says, well, you've published that paper, how do you know what to do when you go to work on Monday morning? It is that science is this necessary pull for all of us into that next stage of the unknown. And the reason that during the time of kind of discord before major paradigm shifts are made is that we can't be heuristic. We can't think what the next thing is because we haven't quite got the last thing right. And all of that, I believe, Janine, is the cauldron in which we are now, 500 years from the time of the scientific method, that great revolution that occurred that really changed everything about the way science was being done. I think it is no small statement to say we are in that same paradigm shift time with the science that is going to be happening as we move forward. So I am going to invite two distinguished speakers to come to the lectern who have developed a series of deep thoughts on the topic at hand today and are going to address the issue of including or excluding sex as a basic biological variable and what the penalty or what the advantage is for that question. And I'll ask each of them to deliver their remarks. We'll take questions at the end because I think that freewheeling conversation is really the thing that we all want to value and have time for. And at that time, we'll try and develop some action plans that we can provide back to the Office of Research of Women's Health on how to move this agenda forward. So the history lesson now being over, the Francis Bacon, repeatable, measurable on a global scale, conciliant, and heuristic are things for us to keep in mind as we move forward through the day. So it's my pleasure to introduce to all of you Peg McCarthy. She is at the University of Maryland, where she is a professor and chair of the Department of Pharmacology. She is a distinguished leader in the area of sex differences and is a noted speaker, has held editorial board positions, and has been on the forefront of ensuring that all of us think very categorically about how to include sex as a biological variable, and we look forward to her remarks. And then immediately following her, Farida Sohrabi from Texas A&M Health Sciences Center, also a leader in the area of sex inclusion for many years, as associate professor and associate department head of neuroscience and experimental therapeutics at Texas A&M Health Sciences. She also is the director of the Women's Health and Neuroscience program at that institution. So two real thought leaders, and I'm delighted to cede the podium to Peg as she comes to deliver her remarks.
Thank you very much, Teresa, and I want to thank Janine for the wonderful invitation to be involved in such an important event, and also Leah Miller for all of her expert help in setting this up. So as Teresa said, I've been studying sex differences for a long time, my entire career, really. I consider myself, rather than a convert, kind of an early adapter, and I sort of now feel like I'm presiding over my own demise. Oh, shoot, I'm so sorry. By trying to encourage everybody else to do what I've been doing, and that's not necessarily a good thing because then you're sort of, I'm going to put myself out of business, but that's okay. Hopefully it'll take until I'm ready to retire. So if we, and I'm a neuroscientist, somebody was asking earlier about other systems. I only study the brain. I study hormone effects on the brain. And so if we think about the brain and we think about all of the many, many, many things that go on in it as it develops, we know that there are sex differences in basically all of these processes. But let's look at what we have learned. So let's just take something as fundamental and simple as synaptogenesis and synaptic pruning. And if you go to the textbooks will tell you that synapses are formed when an axon comes, zooming by and makes physical contact with a dendrite, and that physically induces a connection between the two involving a whole series of different types of molecules, the efferents, the cadherins, et cetera, like that. And that's going to lead to the formation of a whole bunch of synapses in a relatively random manner. And this is then going to be followed by a very fine-tuned process called synaptic pruning, and that's going to be a use it or lose it phenomenon. It's an activity-dependent process. If you haven't made a strong connection, those synapses are going to die. And this is what the textbooks tell us, and this is how the brain builds and hones synapses. Full stop. There's been no other lesson that you're going to learn until we look at some other brain regions. So I study a brain region called the preoptic area. It's got nothing to do with vision. It's just named that because it's pre to the optic chiasm. And we've been studying synapses in this brain area for quite some time now. And if we look here at the male formation of synapses, there's going to be a very rapid increases of synapses during the parallel period of the first 3 months of life in a human. And then they're going to remain absolutely steady throughout life. There will be no pruning happening at all. And because we have measured it at birth, 3 days, 20 days, 60 days, 90 days, it's exactly the same. If we look at females, they're not going to have that rapid increase in synapses early in life because they're not going to have the hormonal milieu. But again, their synaptic pattern is going to stay exactly the same. So it turns out there's more than one way to build a synaptic pattern. It doesn't have to involve pruning. So we shouldn't be stating these global laws of how the brain is built based on a few studies in the many studies in the cortex. Synaptogenesis is also in the textbooks as saying AMPA receptor incorporation is not required for synapse formation. AMPA receptors are a particular type of glutamate receptor. Okay, again, this is in Nature Reviews. This is in the textbooks. But we found in this brain region that, in fact, AMPA receptors are absolutely required for synapse formation. And the other sort of heralded glutamate receptor, the NMDA receptor, is not even in the neighborhood. It is in no way involved in synapse formation in this brain region. So again, this textbook idea that we know how the brain works based on this is not sex bias as much as this is brain region bias. What's true for the cortex must be true for the rest of the brain, which is not true. But we discovered these mechanisms by comparing males and females. Now, the other thing that we discovered, you ask, well, what are different? Everybody nowadays is excited about gene expression. So what are the genes that are differentially expressed in the developing male and female preoptic area? And we did a technique called RNA-seq because it tells you what the genes are expressing. It's highly accurate, although one does use very small n's because it's extremely expensive. So even though it's a mouse, Virginia, or it's a rat, actually in this case, when you're spending thousands of dollars per sample, you keep your n as small as you possibly can. But if you have a big effect size in your sex difference, you can get away with that. So here's a heat map of a bunch of genes that were different in the males and females, just to show the relative expression. And I think that's pretty convincing that that's a sex difference in gene expression. But what was unexpected and interesting is that when we did an ingenuity pathway analysis to look at what kinds of genes are these going to be, well, you think they're going to be synaptogenic genes, they're going to be myelination genes, they're going to be neurogenesis genes. But in fact, what they are is immune cell trafficking, immunological disease, infectious disease here. Again, so we're comparing normal males and females, and we've got all of these immunological genes that are jumping up. So what's going on with that? Well, that actually fit in perfectly with another very unexpected player we had found in the development of these synapses in this brain region, which is that it wasn't dopamine, it wasn't serotonin, it wasn't acetylcholine, it wasn't even GABA or glutamate or anything. It's actually mediated by a prostaglandin, an inflammatory membrane-derived mediating molecule, which was masquerading as a steroid. And that's because, in fact, it's turned on by a steroid. And we worked out a whole series of steps that involved estradiol binding to its receptor, increasing cyclooxygenase 2, and the increase of prostaglandin that leads to the formation of these synapses. Now, to just return for a minute again to the synaptic pruning, which is again just sort of a bedrock of neuroscience, one of the most exciting studies in the field, most exciting findings has been the idea that there's another cell type in the brain called microglia that are in fact mediating the pruning, that the activity-dependent loss of synapses involves their active engulfment by these phagocytic cells called microglia. And this was done in 2 different laboratories, Beth Stevens at Harvard and Cornelius Gross at EMBL in Europe. And they both, Beth looked at the visual cortex and Cornelius looked in the main cortex, prefrontal cortex, I think. So what are these microglia? Microglia are they're the immune system of the brain. They are actually modified macrophages. They're non-neuronal in origin. They come from the embryonic yolk sac. They migrate into the brain early. The blood-brain barrier closes around them, and there they are. They're forever resident in the brain. And they will both respond to and produce prostaglandins. They also show these huge morphology changes from amoeboid and activated to ramified and thin. And one of the things we looked at is say, well, what do the microglia look like in this brain region, and how do they relate to synapses? And we found that the male had much more angry, blebby, amoeboid-like microglia. We also showed that they produce more prostaglandins, et cetera. And more importantly, what we showed is that they actually participate in the formation of the synapses in this brain region. So here is like a little video that shows you the whole cellular process of testosterone comes in from the left. It's aromatized into estradiol, binds to the estrogen receptor, which goes to the DNA and transcribes the cyclooxygenase 2 gene, which leads to the production of PGE2 from arachidonic acid. Notice our microglia just changed their morphology. We also activate the EP2 and 4 receptors, which are adenylate cyclase linked, which are going to activate PKA and phosphorylate subunits of the AMPA receptor. That prostaglandin E2 is not going to activate the astrocytes as well, and in a calcium-dependent manner causes the release of glutamate, which is going to activate our clustered AMPA receptors, leading to the formation and stabilization of dendritic spines. Thank you. Would you like me to repeat that? But the microglia here are an absolutely critical partner in this process. And so instead of microglia pruning synapses, they are actually essential to establishing synapses. So again, we can't have brain region generalizations, and we discovered these things by looking at males versus females. So let's go back to our general model of brain development and look at another phenomenon, neurogenesis, which is here listed as occurring early, but we all know there's a great deal of excitement now about postnatal neurogenesis, in particular in the adult brain, but some people have been ignoring that in the very immature brain, there's also a period of active neurogenesis. And what we have found is that in the amygdala, the newborn females have more neurons and astrocytes than the males, versus in the hippocampus, the males have more new neurons than the females. So what mediates these sex differences? Well, in the adult, neurogenesis is promoted by depolarizing GABA, but in the neonate, we found something completely different. Larry already introduced this this morning, the endocannabinoids, the brain's own marijuana, another lipid membrane-derived signaling molecule. And what we had found is that this signaling molecule is twice as high in females as it is in males, because they have less of the degradative enzymes and more of the FA and MHEL and more of the anandamide and 2-AG. And so you say, well, endocannabinoids modulate GABA and glutamate, so it still must be mediating neurogenesis via neurotransmission, right? Well, no, the answer is wrong, because returning to our microglia, we noticed that our microglia had these big cup-like structures on them. What these are big phagocytic cups. And here's just another view of these are giant, compound, hungry microglia, like the old Pac-Man, remember? And here's this is a nice Baltimore lacrosse microglia. And here we have our Mickey Mouse microglia. This is actually a triple complex cup. So the big question is, what are they eating? They're actually eating other cells. We can see this here by staining them with DAPI, and also a marker for a degradative enzyme that shows they are, in fact, engulfing and consuming other cells. You can see there's this one here is two-fisted, and some of them have multiple cells inside those phagocytic cups. And so it turns out that sex difference in cell genesis is secondary to the endocannabinoid-induced microglial phagocytosis. So here you can see the females have about twice as many new cells as the males. If we masculize them with testosterone, as Patty Hearn was talking about earlier, we can actually block that. But if we block this phagocytosis chemically, we actually increase the number of cells that survive in the male. And that's because the microglia are actually engulfing living cells in the male brain. So when we return to all of our processes that are occurring in the normal brain, we have to add a new one under neurogenesis, and that is murder. He was just a boy. So what about the last process here that's a naturally occurring one is apoptosis. And if we look at our classic views on apoptosis, it's all about target-derived signaling molecules that are sent back to keep the number of cells innervating them alive that they want. But we've known for a long time that the sexually dimorphic nucleus is a naturally occurring model of apoptosis. This is where males and females start with the same number of cells, and in response to estradiol, they are going to die off in the female because she is deprived of this trophic factor. So this is a naturally occurring model of apoptosis. It is not at all dependent on a target-derived neurotrophic factor. It's in fact directly on these cells. Right next door in the AVPV, which is also a reproduction-related nucleus, estradiol does exactly the opposite, and it actually kills cells. And it kills cells selectively based by mechanism, based on their, your guys' internet is really excellent. Based on their phenotype, so that we kill dopamine neurons via caspase-dependent cell death, and we kill GABA neurons via a TNF-alpha trip mechanism. And so it's two different mechanisms by the same steroid are killing off different cells at the same time. This is a fantastic model. Why aren't we using this to understand cell death in the brain? One laboratory in the country was studying the TNF-alpha-induced cell death. I don't know that they're funded anymore. One laboratory in the country was studying the caspase-3 cell death, and they've moved on because reproduction is so underfunded. So why has the value of studying sex differences not been widely embraced? It's in part our fault as a field, and it's just historical, and it was been referred to historically by Larry earlier, that we discovered in 1959 that gonadal hormones permanently influence the developing brain, but it was all based on lordosis and mounting behavior. And so we developed a dogma that sex differences in the brain are determined by hormones, are only relevant to reproduction, and they are restricted to a few brain regions. But there's been three major challenges to the dogma recently. One is that Art Arnold, who's sitting here, single-handedly wrestled to the ground the hegemony of hormones and has said that chromosomes actually matter. Larry Cahill has been very vociferous in reminding us that sex differences in the brain are pervasive and multifactorial. And then sometimes the person who's not here, DeVries, has proposed the notion of sex differences are not always a sex difference. Sometimes the sexes are trying to be the same. So understanding sex differences benefits both sexes, and if we just sort of return again to this idea about the male brain using these immune mediators, which I could give you a lot more data on but don't have time for, say if we look at disorders with developmental origins, disorders with microglia or immune involvement, and disorders with sex biases in prevalence and severity, we can clearly, easily converge on autism, schizophrenia, and Tourette's in just the neurological realm. There's probably more in the other realms, but there's many, many other overlaps as well. So this is a great wedge to give us tools to understand diseases that affect both males and females. And I know that many, many, if not all of the NIH institutes recognize sex differences, but these are the ones that I know of personally from experience or from others' experiences that have now for many years supported sex differences research. But it obviously has not been enough. So it's great now that we're going to try to get something much more systematic and across the board to advance this agenda. So thank you, and then I'll just hand over here to Farida, and I hope that the mail doesn't come up.
You have to accept it.
Oh, well,
Yes.
but then it'll start dinging my mail. You
That
don't want,
is a problem.
and then everybody will see my mail.
Everybody will see your mail. Who wants to see her mail? Okay, that was a great talk. So I'm Farida Sorabji, and I'm very happy to be invited to talk over here. I'm delighted we're having this session today, so thank you, Janine, Susan, Leah, everybody who organized this. And I have to say that I'm standing here with two strong emotions. The first one is a feeling of bewilderment. I did my graduate training in a songbird zebrafinch laboratory. My mentors were actually Art Arnold's protégés. And for 5 years, I did nothing but live, breathe, and think about sex differences in the brain. Song is a sexually dimorphic behavior in this species, and the brain is sexually dimorphic as well. And then I moved on to my postdoc, and that feeling that sex differences were all important sort of frittered away. And now, when I'm studying stroke, that the importance of looking at sex differences is back. So I think I've made sort of a full circle, and I'm very relieved we're having this discussion today. So you're going to see what looks like a bunch of seminar slides, but I'm going to use them to make two important points. And that is what happens if you don't include sex as a variable in your research. And I'll give you one example where by not including it, I would have been down the wrong path with an interpretation. And the second is where we actually exploited the sex difference, and that gave us a very nice window into a therapeutic target for stroke. So with that background, I'm going to use a slide which is a variation on something that Patty showed us, but it's a little bit different. What you're looking at is a male-to-female prevalence ratio for stroke. And the big point I want to make here is that in the younger years, women tend to have far fewer strokes. They're very protected from strokes. And as you get up into the older years, that protection disappears. And so, in fact, towards the age 70s and 80s, women are much more likely to have a stroke, which is unfortunate because that is the window where most strokes are going to occur in any case. It is a disease of the elderly. Even in the earlier time points, some new data from Amy Tofighi's lab suggests that there is already a doubling in the in the prevalence of stroke among women as compared to men, even in that early age bracket around 45 to 54 years. Okay, do you mind? Okay. If you see Peg's email flying through, just be courteous, ignore it. Unless it looks interesting, and then we'll take a quick look. Okay. So in this age group here, 45 to 54, there is a doubling in the prevalence of stroke risk in women as compared to men. This is very interesting because I'm sure, as most of you know, this is the age that pretty much brackets the menopause. And that was basically what brought me to stroke, because of my interest in how ovarian hormones, especially estrogen, might be neuroprotective for the brain. And so that's what brought us to stroke research. The other important sex difference is that not only is there a difference in prevalence, there's also a difference in outcome. That is, among males and females, females are much more likely to report disabilities 3 to 6 months following stroke, as well as out to a year. And these include disabilities with many of the activities of daily living. So 2 points. The prevalence is worse in women at these older demographics, and that the severity of stroke tends to be worse in women as well. And this is an important point because in our animal model, we can't really mimic risk. What we are really mimicking is severity. So typical stroke models, you want to occlude a major artery, usually the middle cerebral artery, and you can do it in various ways. Either you can electrocoagulate it, that's a permanent ischemia. You can put a filament in there and remove it, that's a reversible ischemia. Or you can use a vasoconstrictor, which is also a reversible way of inducing a stroke. In any case, what you end up with is brain tissue that looks like this. This is the portion that is lost to the infarct, and therefore most laboratories will report infarct volume as their measure when they look at stroke therapies or sex differences or age differences. And I'll be using that a lot to demonstrate our data. The other important thing that we wanted to do when I was starting to look at the effects of hormones on stroke is to look at a population that was closer to this older female demographic as compared to the groups that were normally being studied. And most of these studies are done usually in young adult females, if you're looking at estrogen. We moved to an older group, which was a middle-aged group. And in the Sprague-Dawley rat, and I should mention this is true of the rat, not of the mouse. The Sprague-Dawley rat, this senescent female, has undetectable levels of estradiol at 10 to 12 months, has low levels of progesterone, and an elevated level of FSH. So in many ways, the hormonal profile of a postmenopausal female. And so that's the group that we were interested in studying. And what I'm not showing you is a lot of work we did looking at the effect of hormones on stroke in this middle-aged population. And unlike other groups, we found that estrogen treatment to this middle-aged group was actually not protective. It was toxic. So there's quite a lot of controversy. A few labs have shown protection, and a few labs have shown neurotoxicity with estrogen in this older population. The other thing that is different among the young females and the middle-aged females is the low availability of this very powerful growth factor, IGF-1. And IGF-1 has been shown to be neuroprotective in other injury models, and so we took a look at them. And what we hypothesized is that it's likely that the fact that these middle-aged females that have larger infarct volumes have low levels of IGF-1, and that's the reason that they have the poor outcomes from stroke. So we tested that. We gave our middle-aged females either vehicle or IGF-1, and you see a marvelous neuroprotection with IGF-1. So we concluded that this age-related loss of IGF-1 underlies the severe stroke outcomes. And here was one of our perils, and that is we had not actually compared the males at this point. And when we included the males, we found something very interesting. This is infarct volume in adult and middle-aged females, adult and middle-aged males. What you'll notice here is that the adult females have the best outcomes. Several laboratories have shown that. Aging females don't do as well. Interestingly enough, adult males have a worse outcome than adult females, and middle-aged males don't differ from adult males, but they're both have equally poor outcomes in terms of stroke, of infarct volume. So we looked at IGF-1 levels, and if we were right about our hypothesis, then clearly these two groups should have very low levels of IGF-1, just like our middle-aged females did. And it didn't. It wasn't true. In fact, what we found, we were able to confirm that our middle-aged females have less IGF-1, and and giving them IGF-1 will improve stroke outcomes. But if you look at our adult and middle-aged males, they actually make a huge amount of IGF, both in circulation as well as intraparenchymally in the brain. So it's not just a question of IGF-1 availability. What we should be looking at is what is it that the IGF-1 is doing in the female brain as compared to the male brain. So we're backtracking now, and we're looking at what are the receptors that IGF-1 engages, and how do they collaborate with the hormone family of receptors, because it's fairly well known that the estrogen receptor and the IGF receptor homodimerize and result in survival and angiogenesis. So when the ORWH request for supplements came out, we must have been the only laboratory in the country who requested a supplement to add males to our study, which we hadn't done in the previous. The title of my grant, to be in all fairness, is Neuroprotection in the Aging Female Brain. We were well within our scope there. So that was one example of where not comparing sex differences would have badly and incorrectly altered our interpretations. It's not just the availability. There's clearly some other factor that IGF-1 is collaborating with, and that is sexually dimorphic and age-related. So we're hotly pursuing that. The second thing I want to, okay, that was my big punchline. The second point I want to make is the missed opportunities. If you don't look at sex differences, you might not identify therapeutic targets that are just waiting to be seen, in a sense. So we've been working very steadily on the idea that because there are age-related differences in stroke, and we found that they're refractory to hormones, that perhaps that there are somewhat larger mechanisms at work here, perhaps epigenetic changes that occur with aging, and we're looking at the epigenome of certain cell types in the brain, and we're also looking at a class of non-coding RNA that exert a wide effect on the transcriptome. And so using these two, we've identified a microRNA, and I won't go into the details of how we identified it, but needless to say, we were very convinced that it would be neuroprotective. And we did that experiment. We felt that IGF, at LET7F levels, the higher the LET7F levels, the worse the outcome from stroke. And so if we had antagonisms to this microRNA, we would be able to see neuroprotection. And sure enough, we did. We did this experiment in adult females. We gave this group scrambled oligos. We gave this group antagonisms to LET7F, and we saw the most profound neuroprotection that I've seen in any of the treatments that we've tried so far. Being very excited about this, we moved on to look at males. Absolutely no effect. We looked at ovariectomized females. absolutely no effect. It was starting to dawn on us that perhaps there was an interaction with the hormones. And then we did the group that we really care about, which is the middle-aged female group that has the higher risk of stroke and the worst outcome from stroke. And lo and behold, we saw a slightly toxic effect. So we were back to the drawing board. And it was at this point that it dawned on us that we should be using the biology to actually identify therapeutic targets. And let me tell you what I mean by that, okay? Oh, I just wanted to point out, I know this slide has been shown repeatedly. Louise is going to be so thrilled. The idea of sex differences in outcome, and Patty, to be fair, Patty's a senior author on this paper. There are sex differences. What she's showing is an absolute reversal of a neuroprotective agent in females as compared to males. And in fact, in her minocycline work, she showed the opposite of what we did, and that is minocycline is very effective in males and completely ineffective in females. Okay, so here's where we started to think about we should be exploiting these age and sex differences to develop therapeutic targets. Here's a picture I've already shown you. This is stroke outcomes in our young and middle-aged females, young and middle-aged males. And you can tell that this young female group has the best possible outcome. So what we need to find is what is this group making or not making that we should be treating our older female group with, and possibly the males. And of course, the most obvious one would naturally be estrogen levels. Estrogen levels are usually high in this particular group. It's been tested, as I pointed out, but there's been anomalous effects in an older population. So our question was, what else would have the kind of comprehensive effects that estrogen would have on multiple body systems? And that's when we moved towards epigenetics and alterations in non-coding RNA. And here, we did a huge panel, and I'm only showing you one slide. We looked at circulating microRNA, brain-resident microRNA following stroke in all these populations. And as a neuroscientist, it was somewhat disheartening to find out that the brain was actually the least promising indicator of biomarkers for stroke. In fact, the best biomarkers were actually in circulation, in serum. We found one particular microRNA biomarker. We repeated this whole experiment a couple of times, and this one withstood all sorts of tests. Here is our stroke data, okay? Adult females, very, very good outcomes. Middle-aged females, adult males, poor outcomes. And this is microRNA 363. It is highly expressed. Just to be sure, when you show data that is DCT, it means that fewer cycles were needed, and therefore there's more of this particular microRNA. There's more of 363 in the adult female circulation as compared to the middle-aged female and the adult male. And this, we thought, would be the target to look at, because now we had a marker that discriminated our good outcome group from the poor outcome groups. So what the most recent experiment, and this is unpublished data, is we used microRNA 363 mimetics in our middle-aged females to see if we could improve outcomes. And we did. We see a very nice protective effect. What I'm showing you here is infarct volume is improved by 363 when given to middle-aged females after stroke in circulation. And what I'm not showing you is the behavioral data, and we also saw behavioral improvement in the mimetic group as compared to the scrambled group. So those are the two examples from my work where looking at sex differences paid off in a very good way. And I just want to end with the last slide, which I titled Random Thoughts because it occurred late in the evening and wine was involved. So I think even among those of us who agree that sex differences are important to study, there appears to be a difference in how much and how to execute this. And here's where I come down on this. It is expensive, it is time-consuming, but I think not looking at it is poor science. One of the examples that I've made to a couple of my colleagues over here is the idea that the NIH should pay us more to include sex as a variable. May be a good start, but that's not what we should be thinking about. We need to inculcate a mentality that including sex as a variable is good science. I wouldn't think of an investigator telling the NIH, if you give me the money, I'll do a control group. Or if you give me the money, the pharmacologist saying that I will include all of my doses. All good pharmacologists will do a huge range of doses. It doesn't matter how much money you give them. That is where we need to get to with this idea of including sex. females are not more difficult to work with. Now, this may be a little bit biased because I've been working with female animals practically my entire career. Yes, there are considerations that you need to pay to the estrus cycle. They won't affect all outcomes, and if they do affect your outcome, there are ways to control it like you would do any other variable. Yes, it does include more animals, obviously. And the big point I want to make is that this is a wonderful opportunity to find out mechanisms. Sex differences can teach you something about mechanisms. It can get us to start rewriting textbooks, as Patty mentioned earlier, that the cell death pathway was an entirely male-determined pathway, and that's something that needs to change. It's also a great way, if you see sex differences in your particular disease, to try and find a therapeutic target that you can address. So there's a pragmatic approach to it. Thank you for your attention.
Thank you. We couldn't have selected better speakers for this session, which is to talk about the impact of including and not including sex as a basic biological variable. And I think what you've heard are really important summaries that there's a real opportunity for discovery, and we really are in a time of a paradigm shift where we can rewrite the textbook. That if we look at cost, we can really reframe that. One of the issues that was brought up was the relative cost of microRNAs, but as we learned from Francis Bacon, we have to be able to have multiple ways to look at a particular endpoint. So not just one endpoint gives us the outcome. And so that will mitigate some of the cost as we move to engage sex as one of the variables. And that sex, as was well said, is equivalent to dose, time, temperature. It's a testable variable that needs to be included. And that some of the discoveries that are occurring, for example, the nitric oxide synthase, if that was actually transferred across to other disciplines, for example, in peripheral vascular disease, has just learned that nitric oxide synthase works differently in males and females. And so for peripheral vascular disease, particularly as it relates to diabetes, there's been a lot of lost pace and lost money because we didn't know that early enough. And now that work is just coming out of the peripheral vascular disease. And so the opportunity for discovery and the missed opportunities, and maybe better said, the delay in those opportunities that can be hastened if we include sex as a variable, I think were 4 takeaway points that I took from what you said, which was really exceptional. So now let's open the floor for a discussion on both the science that was presented as well as the larger, more macro topics of what the opportunity is for inclusion of sex in basic science and translation. Yes.
This is just a sort of a general comment. You started off by reiterating the comments of Francis Bacon, and one of the tenets was that it had to be globally applied. We've just heard 2 elegant talks about how these mechanisms differ and how we're talking the whole conference about sex differences. So these are 2. This is where it can't be globally applied.
Well, so
let me just finish.
Yeah, measurable on a global scale.
So what we have to think about is how we define that globally applied, because we heard from Larry and some of the others in drug development that it stops when it goes to the application of a therapeutic, because clearly these 2 mechanisms are different. A drug may not work the same, and we have the Ambion as a real example. So I think we have to be careful now going forward that we know these differences are important to physiology, potentially to diagnostics and therapeutics, how we interpret that word globally applied.
Yeah, and so Bacon is actually measurable on a global scale. I'll have to go back and get the original, but I think that's what it is. So I think it's a different concept than the way you presented it, although both are concordant with discussion points that we should have. How do we make sure that we don't generalize, that we become more articulate in personalizing? And personalizing is individuals, how the FDA can look at males and females and discriminate drugs that can be good for one or the other, Ambien, the example that was given earlier, and many other drugs. And then how can we look at different parts of the anatomy, the brain being a beautiful example of anatomy that can change by microns in terms of mechanisms, and make sure that we have the scientific method in place to be able to describe things precisely. And I think the third, as we macro back out, then how can we take something that might have been learned in neuroscience on nitric oxide synthase, which I think is what you're picking up on, and it's now true in peripheral vascular disease. And so it might not have been true in peripheral vascular disease, but how can we as scientific practicants take each of these different scales and be able to use and adapt them appropriately so that we make sure that we hasten science and don't miss opportunities and don't fool ourselves in the process. Larry?
Just to build a bit on this question of individual differences in personalized medicine, first I want to say those are a couple of great talks. I really enjoyed them immensely. I'm going to have to watch the video to relearn everything and have it consolidate in my head. But this is a question now for you and really for anyone. And on this topic of individual differences, I struggle to understand, as our field rightly embraces personalized medicine and individual differences, it seems perfectly logical to me that sex influences should be leading the charge. And it ain't. I've talked to a lot of people who actually try to use individual differences to make sex differences go away, which is itself kind of silly. But I would love your thoughts, the thoughts of everyone on why is it the case that this thing that should be leading the charge of this great, excellent movement towards personalized medicine has really been relegated to sort of poor cousin status.
So I too feel that for personalized medicine, this should be the bottom rung of the ladder that we should be looking at. But when you think of it in the context of what has actually happened, it's not surprising. For example, it took us years to actually realize that children are not little people. And it's simply just decreasing the dosage down isn't going to help. We're still struggling against that very, very basic idea. So the idea of individuals that are tall and may have some differences in weight and muscle mass, but have the same general organs, surely there should be no differences that we need to worry about. I think in the scale of differences, the sex difference story is certainly a bit newer than the age difference story is. And I feel like we have to accelerate that. We can't just wait for the mainstream to naturally come to it like they came with the children's drugs. And that's my take on it.
So I guess I'm going to take a little bit of a different angle, but partly this from working in the medical school environment. I think one of the issues of why we haven't been treating gender as personalized medicine is because of gynecologists, because we have a doctor for your femaleness. Right, we don't have there are proctologists, but men don't see their urologist. Every 6 months. They don't have to go to have their Pap smear. Women health involves a specialized medical discipline, and until we start to, and some of the HMOs and things are getting more visionary and starting to move the gynecologist into the woman's general practitioner, because she can also just prescribe you a hypertensive drug or treat your diabetes or et cetera, and give a holistic care. I mean, internists out there now act like psychiatrists. They prescribe more antidepressants than the psychiatrists do. So until we stop this balkanization of women's health being your uterus and your ovaries and that's it, I think that's part of the problem.
Just to rephrase that slightly, I think that's a great point, that people think that this issue has been taken care of. It's over there in bikini medicine, and it's done. So we don't need to worry about that.
I think it's maybe even more negative than that. We've heard the terms even today of anti-American and witchcraft. So that takes it even beyond. It's just been specialized, so therefore we don't have to think about it. I think there's a macro discussion here that there's something different about this women's issue. And women only came into science in numbers in the 80s. Women weren't on study section until a women's group from AWIS had to sue the NIH directly to be able to have women on study section. So I think this is part of a larger macro issue, and I think it's just dead simple to imagine sex, if we could take our lenses off some of these other political issues, as just equal to time, temperature, and dose. But the fact that we can't means that we can't get to personalized medicine by simply splitting the population in half. And it means a whole circulating additional kind of issues surrounding women in the workplace, women's health. And the fact that men haven't brought this in to be internalized, this is their issue either, I think is really critical. And I'll say one last thing, and that is if you looked at the 60 Minutes report online, what was the ad? Viagra, 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 Viagra. It's still Viagra. And, so there is this complex psychology that is also a part of this. And if you use terms like witchcraft and anti-American, whether you're male or female, about this issue, it says that there's something bigger about this that we have to grapple with as a field. And we have to get more articulate in describing why this is valuable just as a base biology, dose, time, and temperature.
Can I just add to that? So I think one of the other side of the coin about the point that you made is that we need to stop having this discussion only about how women have been disadvantaged by medical research not taking into account female populations. There are good examples of that, and we need to bring those to the forefront too. So virtually all of the data on osteoporosis, all the normative data is based on women. So they're treating men based on women's normative data. The same thing is true for breast cancer. Breast cancer is women rarely get to the point, if they have access to a health center or a good health care, to get to the point where it is discovered at the very last minute. But for men, that is usually when it is discovered, is when it's metastatic. So there are equally good examples about how ignoring sex differences disadvantages both populations, and that we need to give voice to that part of the story as well when we talk about women's health.
Those were great talks, guys. They were wonderful. So I think in answer to your question, I think it comes from the fact that when you look at drugs, that in general they work on relatively equally in men and women. So if you give someone who has hypertension, you give them an antihypertensive, and the differences can be subtle but meaningful, especially in a large population. And I think if we do more investigation in collaboration with the FDA, and we look more at pharmacokinetics and pharmacodynamics of drug action, that's what's going to help make this argument that's going to really convince people. But we haven't really looked at that. And you have this one case of Ambien, but there are other cases that really haven't made the news. And I think that's where we can get the ammunition.
Point. So bringing FDA to the table would be a good next workshop, Janine.
I thank you. Those were great talks. I wanted to just respond to what has been brought up in terms of the OBGYN community, if you will. I just returned from the North American Menopause Society, which is really very well populated by OBGYNs. And I think there is a great realization there, even though it may not be as explicit as we're talking about today, sex differences in biology. There's a great appreciation for sex differences. And one of the comments, to pick up on what you were talking about, that 20% of osteoporosis cases occur in men. And so, and there is this idea that we really need to be studying men and women together. And so I think there is this awareness. It's just maybe more implicit than explicit. And then I just wanted to mention something about your wonderful Venn diagram. And your talk was terrific. I loved it. And you had the mental health issues that were illustrated in that Venn diagram. And I want to also just mention that there's a huge dimorphism in sleep and pain issues in women that overlap markedly with depression. And it really behooves us to study a lot of these issues together and not in isolation.
I think we would be remiss to leave this session by vilifying any one discipline. So OBGYN is terrific, and perhaps to pick up on what Larry Cahill said earlier, that education at the medical school and basic science level is so important so that women's health and men's health is adequately represented across all the spectrum of disciplines, so that gynecologists can take care of women's reproductive tract health, which is an important discipline. And so it should be the breadth of medicine that understands how to manage all of us as individuals.
I wonder if we might gain more traction if we start thinking about sex differences within the scope of individual differences. At least at the level of the hypothalamus, sex differences put males and females in two categories. But for many other disorders and functions of the brain and body, it's actually a much more normal distribution, and you have, if you will, female-like and male-like prototypes, but you'll have, as the overlap of the distribution shows, you'll have males that respond more like females and females that respond more like males. And we need to be thinking about how we don't talk about just a male this and a female that, and a way to think about the descriptions that aren't necessarily gendered.
Yeah, I couldn't agree more, and I think that it's just like we have to be careful about sex and gender. We have to be very careful about sex dimorphism versus sex difference versus sex influence. And there's way too much abuse of the term sex dimorphism, which means di-to-morph forms, and it means really separate. And right, most of what everybody's interested in is either sex influence or sex difference, with huge overlap. Absolutely.
Comments for the speakers? Well, I think then we can cede the floor to the next speaker. Thank you, Janine, for inviting this session.
Well, it's time for lunch. So we'll reconvene at 12:30 sharp. Cafeteria is right across the hall, and for those online, we'll be back.